welcome to Module 10. This is on the nervous system, in my opinion, the absolute best system in the human body. Very interesting system. We'll take a look at all of these learning objectives today. You could use these for your studying. First, we'll start off with the organization of the nervous system. What we need to know is that the nervous system is split into two different big categories. You have the central nervous system, which will often use the abbreviation CNS, central nervous system. As you could imagine, it is the most central part of the body, right? Look at where it is. It's in the center of the body, whereas the peripheral one is on the side. So central nervous system is centrally located. It consists of the brain and the spinal cord. People often don't think of the spinal cord as the central nervous system, but it is very important. And the function of the brain and the spinal cord, this central nervous system, is to, number one, receive information. Information comes up, we'll see, through the peripheral nervous system and enters into these areas. So it receives that sensory information. Then it integrates. Integrates means to bring together and piece together all the sensory information, because the sensory information is very specific and parsed out. Maybe a little touch to the finger here, or a little pain to a little receptor on your skin over there. It's very specific for what it's picking up, and the brain and spinal cord need to integrate together all of this info so that it makes sense, right? All these tiny little pieces together don't come together unless there's this central nervous system. So all sensory info will kind of come together in the brain. And the brain has some areas where there's consciousness, memory, emotion. So it will kind of add those aspects to the sensory info coming in, right? Maybe you'll end up piecing together that you see a bear in front of you. You might hear people screaming. You might end up seeing that it's dark out and feeling cold, right? All those senses come together. Your memory will say, oh, I've watched a movie where that happened and then they died, right? And so your emotions will kick in. You'll be scared. You'll want to run away. You're definitely going to be conscious. So this central nervous system is complicated, very complicated. And it takes in this simple sensory info and it thinks about it, integrates, thinks about it. Then you don't just do nothing once you see the bear, you feel the cold wind, you hear screaming. You actually do something about that. Your central nervous system will then coordinate motor outputs to execute a response. So they will, once they've processed all this information, they will send down signals we call motor outputs to execute a response. Maybe contracting your skeletal muscles to make you run away from the bear. Maybe contracting your muscles in your face to allow you to scream, right? All of these are going to be what the central nervous system does. So it receives info, thinks about it, and it makes a decision on what to do. The peripheral nervous system, also called the PNS, peripheral means on the sides. So we say a horse has peripheral vision, right? It can see on the sides. That's because, so this peripheral nervous system is the nervous system that's on the side away from the center, right? Do you see all these spaghettis that are coming out, right, from this middle section? All neural tissue found outside the brain and spinal cord is part of the peripheral nervous system. It's the outside from that center, right? This includes sensory receptors, like at the ends of your fingers or on your skin, you have little receptors that detect things like the touch, or detect photons in your eye, right? So sensory receptors They're nerve endings. They're usually at the end of peripheral nerves. Sorry, or at the beginning if you want to think of it that way. So these are all peripheral nerves, okay? Because they're nerves that are on in the periphery, on the outside, right? They're on the outside away from the midline. And these peripheral nerves and the sensory receptors, they do very simple work. I want you to think of them as very basic, right? So they carry sensory info to the CNS. Some of these nerves just, let's say this little nerve here maybe just detects touch in this area of the finger, and bzz, bzz, it sends that, hey, I just saw touch in this area of the finger. It sends that up to the spinal cord and or the brain stem. Maybe this little nerve here says, I detect pressure in this area of the thumb, right? And it sends that info up. It's very simple sensory info, like very specific as well. And then your central nervous system pieces it all together and brings all that information together, which is very cool. So the PNS, it's really just carrying sensory information up to the central nervous system, the brain and or spinal cord. And the second part, once the brain, remember I told you that the brain and the spinal cord, sometimes they coordinate motor outputs. They decide, okay, now that I see this, I'm going to go and move my muscle. Well, that signal will go down the brain, down the spinal cord, and it needs to go and contract your muscle. So half of these nerves, right, coming out, they're actually carrying information in the other direction down to a muscle or to a gland. And they will actually contract muscles so that you can move your skeleton, right? So they carry motor information out of the CNS to we'll give the easiest example to muscles so that you can contract them and do something, right? Learning about the nervous system is very interesting because you realize how much more robotic the nervous system is than what you think it is. We are in humans that respond to stimuli, and then so a stimuli comes in and then we act upon it, right? So again, I'm going to merge the two together. Information from the PNS, let's say someone hits you over here, right? Your best friend hits you over here. These little neurons will sense pressure. Many neurons in this area will sense pressure and signal that information up the PNS to the spinal cord, up to the brain, right? And then your brain is going to integrate memory, emotions. Oh, I know that my friend is just bugging me. Emotions, I know that they're jokey. I want to bug them back, right? So I want to punch them back. This is getting violent, sorry. And then the brain will put that output of please go punch that person back, and it'll send that neuron down the spinal cord, out the PNS, out a peripheral nerve to maybe this muscle here in the biceps muscle, so that it'll contract so that you could lift your arm. And then some muscles in your hand, there'll be some neurons that are send information down so that these muscles will contract to make a fist, and you will end up having a response, right? Remember homeostasis. You have some stimuli that initiates, you sense something, a receptor, you send it up to the control center, and then you send a response down to the effector. This is the nervous system. This is the master control center of the body. So very cool how there's these two divisions. They work together, obviously, but there's this central part and peripheral part, right? Half of these nerves are sending information up highways, and the other half are sending information only down the highways, right? So some are sensory, some are motor. Sensory, meaning they're bringing up information. Motor, they're going down to a muscle to do a motor command to actually move, right? It isn't so easy as that, right? So we have our CNS, our PNS, but there's different ways of dividing the nervous system. We can also divide it in terms of sensory versus motor divisions of the nervous system. And I really like this diagram. I know it looks complicated, but over here you see the central nervous system, the brain and the spinal cord in this gray, and the PNS, the peripheral nervous system, all these yellow spaghettis coming out, right? And so what I want us to do is just to look at how information either goes up to the CNS, to the spinal cord and brain, or how it goes down. When it goes up to the brain and spinal cord, right? When these little spaghettis bring information up, this is called the afferent division. A is for arriving, right? It is arriving to the brain. So we call it the afferent sometimes, the afferent division. It's going to the brain and spinal cord. It sends sensory inputs from the PNS to the CNS. So look here, we take these little spaghettis, it goes up to the CNS, to the spinal cord and or brain stem. So we're sending info up to this CNS over here. Now there's two divisions of afferent information that goes up. There is what we call visceral sensory receptors. Visceral means your viscera or your visceral organs are the organs inside of your body, right? Think of your intestines, all your stomach, all of those inner organs that are inside, right? That's visceral organs. So visceral sensory receptors are on the ends of the neurons that monitor your internal organs, right? So this division is for things that are more on the inside of you, right? You actually do monitor information inside of you. Think of it this way. Sometimes your brain needs to get signals. Maybe you're not aware of it. You're not conscious of it. A lot of the internal things in our body we're not conscious of, but our brain definitely is does get that information. So, for example, an example of a visceral sensory receptor is on your stomach. It feels the stretch of your stomach when food goes in to tell you how full your stomach is. Right now, I can't ask you to determine that. So it's not conscious. It's not in the upper parts of the brain, but you definitely can determine. You're sending information constantly about that. About how inflated your lungs are. There's information being sent up to your brain about that constantly. About your heart, about how full your bladder is, right? And actually, eventually, it does become conscious when it's too full. But there's always information being sent up, not contracting your bladder, not contracting your heart chambers, not deflating your lungs. I'm talking about sensing things, picking up information and feeling the pressure, right? You sense things, you don't necessarily do anything in response. That's the efferent division, to do something about it. The afferent is to pick up information, so to monitor the state of internal organs, to monitor the blood pressure. There's sensors around blood vessels that will detect how much blood is flowing through it, through pressure. So that's your visceral sensory receptors monitor internal organs. On the other hand, there's another branch of neurons that are called somatic sensory receptors. These monitor skeletal muscles and the skin, right? So when I touch any part of my skin, when I feel pain on any part of my skin, when I feel pressure, when I feel vibration, when I feel temperature, all of that is part of these somatic sensory receptors, okay? These monitor skeletal muscles and skin, so the external environment. So I like to think generally the visceral system as more of the inner parts, the internal organs. This is not really a conscious thing. We can't right now feel how much our bladder is full. right? Like you can't right now be like, oh, I'm about 35%, right? So this is not conscious, but it doesn't mean the signal's not there. It's being sent to the brain. It's just not in your conscious area. And the somatic sensory, this is often conscious. When something touches your skin or you feel pain or temperature, this somatic sensory receptors, they send neurons up and they go to the CNS, and this is going to be voluntary. We'll see that in a minute. But all this to say, the afferent division, right, is the sensory division that sends things, sensations up to the central nervous system. The efferent division sends motor outputs from the CNS down to the PNS, okay? So it sends information from here down over here. So let's just review our afferent division first. The visceral sensory receptors, they monitor internal organs. Somatic sensory receptors monitor skeletal muscles, joints, and skin surface. They'll send all that up. Then sensory information, all in this division, right? All these little receptors at the ends of the nerves, they send it up these spaghettis, right? So at the ends here, you have the receptors and they send up the spaghettis. Those spaghettis are sensory information within the afferent division. We're going up to the peripheral nervous system, and we get to the central nervous system. This is where the brain and spinal cord will process and integrate information and coordinate sensory input and motor command. So remember what I said the brain and spinal cord does? They receive info, they think about it, they process and integrate it, and they coordinate a motor output, a motor command. They send a signal down through the efferent division to do something about what they've thought about. So motor commands within the efferent division, we have two divisions. So efferent means exiting, right? Here, afferent meant arriving to the brain and spinal cord. Efferent meant leaving the brain and spinal cord. So once a decision is made, it goes down and it leaves the brain and the spinal cord. It will go over here. It says sends motor output from the CNS down the PNS, right? And there's really two divisions that we care about. There's the somatic nervous system, not the somatic sensory receptors from the afferent, but the somatic nervous system. I know it's annoying that they sound similar. And the autonomic nervous system, the ANS, we call it. And so we're going to go through each of them separately. When you send a motor command down from your brain to something, you can either go and do it voluntarily or involuntarily. So over here, some motor neurons of the somatic nervous system, right? Some of these motor neurons go down the somatic nervous system, and they will actually send signals to voluntary effectors. What does that mean? If it's voluntary, it means you do it on your own, you voluntarily do it, right? You have conscious control of it. So for example, if I ask you right this minute to go and contract your biceps muscle, you will go do it. If I ask you to go and lift up your leg, you will contract some of your quadriceps muscles, right? And some other ones that are down there. Any muscle that you right now, this minute, can control is part of the somatic nervous system, okay? Skeletal muscles. I need you to know that this is skeletal muscles. Remember that smooth muscles and cardiac muscles, they're not voluntarily controlled, right? So I can ask you right now to contract your biceps muscle. This must be the somatic nervous system. These are skeletal muscle effectors that you can control. We talked about them in the previous unit where we talked about neurons going to these skeletal muscles and causing them to contract, right? This is what we were talking about. So motor neurons of the somatic nervous system send signals to voluntary effectors and that's pretty much just skeletal muscles. Motor neurons of the autonomic nervous system, look at what the name says, autonomic, automatic, right? Automatic nervous system, I like to call it, but it's the autonomic nervous system. This is things that you are not in control of. Involuntary effectors. Your body will just do this on your own, and you cannot control those things, okay? And there's three effectors that these neurons can go to. So the brain or spinal cord will send neurons down to three types of effectors, either smooth muscle, glands, or cardiac muscle. So smooth muscle, cardiac muscle, glands. Don't really worry about adipose tissue. Forget about parasympathetic, sympathetic for now. So the autonomic nervous system sends down information to these tissues. And remember, so you don't see skeletal muscle here because skeletal muscle is voluntarily controlled. Here you see smooth muscle. Do you remember where we find smooth muscle? Think about it for a minute. We find it in hollow tubes in the body that contract, such as your esophagus, your stomach, your intestines. Right now, if I ask you to contract your intestine or your stomach or your esophagus, can you do that? No. Does it contract? Yes. Involuntarily. Your conscious parts of your brain, right, that give motor commands out, they are not responsible for that. There's lower parts of your brain that are involuntary, and they will send signals to contract them. You don't need to worry about contracting your esophagus or your stomach. It does it automatically. The autonomic nervous system does it automatically. Second thing here is glands. When you get stressed or actually when you're hungry and you're secreting certain hormones, glands contract, right? So let's say you're stressed, you have sweat glands that squeeze to secrete sweat out. Is that under your control? Do you love to sweat and just do it for fun? No, you have no control over your sweat glands. You have no control over any gland secretion. This is the autonomic nervous system. The pancreas has certain glands and they'll squeeze out certain hormones. That is not in your control, right? I can't ask you to do that right now. Your stomach too has certain glands. You don't have control over that. So this is also under autonomic nervous control. That's part of this division, the autonomic part. And lastly, cardiac muscle. Your heart is not made out of skeletal muscle. It's not made out of smooth muscle. It's made out of cardiac muscle. And we said, if I ask you right now to contract your lower chamber of your heart on the right side, can you do that? If you can, you should call us and we will study that, because that would be crazy. Cardiac muscle, right, is not under voluntary control. You cannot yourself go and contract parts of your heart. You can right now lift up your arm and contract your biceps, but you can't contract your heart. So all three of these are under automatic control. You don't need to worry about it. It's a good thing you don't need to worry, right? You're walking down the street. You don't need to worry about pumping your heart every so many milliseconds so that you can pump your blood around, right? So this is actually for a reason that this happens autonomically. Yes. So all this to say, I want you to realize here that there are afferent divisions, sensory divisions that send info up. Some of those things are from the internal organs that are sensing things. Some of it is from your skin that's sensing things. So internal or external things. And then over here, once your brain makes a decision about what to do about that sensory info, it sends decisions down a separate set of neurons. Some of those neurons are somatic nervous system and will control your skeletal muscles. And some of those neurons are autonomic nervous system, and they will control things that so the other types of muscle, like smooth and cardiac, and also glands, which you're not in control of, right? And now we're going to talk about this efferent division a bit more. So the efferent division is split into two parts. We have the somatic and the autonomic nervous system. I want to remind us that this somatic nervous system moves skeletal muscles voluntarily. It actually contracts the muscles and it moves the bones voluntarily. So that is our SNS, the somatic nervous system. Our skeletal muscles contract. And then over here, the autonomic nervous system, remember I said it affects smooth muscle, cardiac muscle, and glands, right? Those were the three things I showed you here. I want you to know that this division, the autonomic nervous system, is split into two parts. There is the sympathetic division and there's the parasympathetic. They all will go to smooth muscle, cardiac muscle, and glands, but they send their own neurons and they have a different response. The sympathetic division will mobilize body systems for fight or flight. We call it, flight or fight response. This S sympathetic division is the stress division. What do we do automatically? We get stressed automatically. We're not really in control of that. And we also relax and are in parasympathetic division autonomically. And that's the two divisions we're going to see. So the stress part of the autonomic division, this sympathetic division here, that helps us when we are running away from a tiger. When we are needing to escape this situation, right? It's our flight or fight response, right? If we were organisms that didn't get stressed, we would see a tiger run and we'd be just staring at it and then we'd get eaten. No, evolution has brought this division of the nervous system because it is good for our bodies to get a short-term stress, not long-term. We'll talk about that later on. So that's the sympathetic division, okay? Then there's this, so I want us to imagine what would happen, right? Sympathetic division. So smooth muscle, those neurons that go to the smooth muscle might stop digesting stuff, right? It's not time to rest and digest. It's time to fight or flight. So they might go to the digestive tract and say, whoa, stop digesting things. It's time to run. They might go to the urinary system and say, whoa, stop. We're not going to be doing that right now. We got to run. It might go to the blood vessels and constrict them to help increase your blood pressure to try to go and run away from this tiger over here. And then cardiac muscle, it'll go to your heart and it will go and try to increase the pace of the heart so that you can run away from the tiger. It'll go to your sweat glands and allow you to expel heat so that you could run away from the tiger and not get heated, right? So a bunch of things we'll see later on happen for the sympathetic division to help you to run away, right? The parasympathetic division conserves energy. So once evolutionarily, right, once the little squirrel got chased by an attacker, then when the situation was over, there's this time for the little squirrel to conserve energy, right? And to calm down and repair and rest and digest, right? It's a evolutionary process that sometimes you want to be in this stress mode, sometimes in this relax mode. We're resting and digesting to heal from any injuries, to get ready for the next attack, to make sure that we're replenished. So this division here is the rest and digest response. This might tell your smooth muscle and your digestive system to try to digest and rest. Your cardiac muscle to slow back down. We don't need you pumping. We're not stressed now. Glands to stop secreting sweat, right? Maybe secrete saliva now to start digesting. So it's interesting that this division has these two branches, the stress branch and the rest branch. In terms of somatic nervous system, we remember I said that this is really the voluntary movement. This we're in control of. It doesn't have a stress or a relax branch. It's a conscious thing that we're in control of. As I said, we're not really in control of when we get stressed or relaxed. There's we can do things to try to help it, but if you see a tiger running at you, your body will go into stress mode on its own. You don't need to ask it to do so, right? If you're in front of a class and you're giving a presentation, I don't think you're in control of sweating, high heart rate, and all of those effects that happen. So that is the divisions of the efferent system. Now I think I've already covered this, but I want to reiterate it because it's such an important point. Remember on lecture 1, we talked about homeostasis. We talked about how you sense things, how you integrate them, how you send a efferent command out. I want to go over that now for the nervous system, and I have already, but I really think it shows us, it's very cool how the nervous system is so robotic. Like, we are processing machines. Like, we seem to have this higher conscious ego thing going on as humans, but we really are robotic. We respond to sensations, we process them, and we send a motor effect command out to do something. And I want us to start seeing the nervous system this way, as this processor. So, number one, right, sensory receptors detect a stimulus. I want us to keep using these words we used in the first lecture. So a sensory receptor detects a stimulus from inside or outside the body, such as sometimes it'll be temperature. In our skin, we have a lot of thermoreceptors. Sometimes it's light. And this is the actually light that's going to be detected in the eyes. So this example here is actually the light. Photons will be detected on the back of the retina. So these afferent neurons here, they have little detectors here for light. Sound when it's in the ear, smell our nose, pressure in blood vessels sometimes, or on skin, we feel pressure, pain on skin, ion concentrations for thirst, blood gases, we detect how much gas we have, oxygen and CO2 in our blood in certain vessels, blood pressure also. So all of these are different types of receptors. Each neuron kind of has their own specialty. So over here, let's say this neuron that's in the back of the eye detects light, right? And it detects light, and there's many little light pieces that come together to when you see this glass of water, right? So you detect all of these, that light and that little pixel of information of that picture, right? This little piece of pixel of this glass goes up. That information stimulates this neuron that says, I saw this little pixel here, right? That will be carried up an afferent neuron, right? Remember afferent? Afferent divisions are the ones that are going up like this. It'll go up the afferent pathway. So the nerves carry sensory information to the CNS, right? It'll go up to this green neuron here, right? I want you to look at how we draw these neurons. Often we'll draw it like this. We'll draw the little branches here. The terminals look like this little over here. So we send info up the afferent pathway. This is sensory information to the CNS. It gets brought to the brain or spinal cord. This is the control center. In the central nervous system, two main things happen. First, there is integrating, right, and processing and interpreting of all that sensory input that came in. So, this little piece of the glass of water is here. It's sent up. This little piece is sent up another neuron. This little piece over here is sent up another. And it's all pieced together in the brain. Secondly, emotions, memory, all of those things are already in the brain, and they get added onto what you're seeing, right? Oh, I know that this is a glass of water. I've drank from one before, and I remember that. And then a decision is made, and a motor plan is sent to the PNS. Let's say this brain says, oh, but you haven't had water in a while. These other regions of the body are saying that you're dehydrated, right? And you see this glass of water here, right? You should go reach for it, right? Do you see how that's integration? It's making a decision and a motor plan so that it can be sent to the peripheral nervous system to do something about that information that came in. So it sends that information down to through a motor pathway, right? So nerves carry motor information to the PNS through efferent pathways, right? If we look over here, this is my efferent pathways down this way. This was what my sensory info, my integrating up here, and then down the efferent pathways here. So Now we're going out the efferent pathways, and we're going to the muscle here, right? And over here, the motor effectors receive information sent from the control center, the CNS, and they execute a response, right? I'll give you an example. In this specific case, right, the neuron, the brain told it to go and grab the glass and lift it up, right? So one neuron would go to the biceps muscle to contract so that you could bring the glass of water up to your mouth and drink it, right? So a skeletal muscle was told to contract. Sometimes, not in this case, but sometimes actually a gland is told to release a substance. That would be through the autonomic nervous system, whereas this is through the somatic nervous system, the skeletal muscle. This is a somatic response. But sometimes a gland might be told to secrete a substance, or a cardiac muscle might be told to contract, or a smooth muscle might be told to contract. So those are all possibilities, right? In this case, it was a skeletal muscle that contracted. But I want you to understand that the motor effector, this is doing something about it, right? Like you're actually doing something, you're contracting, you're moving, thanks to a response that was asked of you. Sorry, thanks to a command that was asked of you, you do a response. So I want you to pause the video here and try to answer this question. Which of the following does not require use of the peripheral nervous system? Pause the video and take a crack at this. All right. So, which of the following does not require use of the PNS, the peripheral nervous system? Raising your hand in class. So let's first of all go remind ourselves of the peripheral nervous system. This is the peripheral nervous system, sending info up. Thinking about the info in the brain and spinal cord is not, right? Sending info down to do something is part of the PNS as well, right? So only the brain and spinal cord, this thinking, receiving info, thinking about it, and thinking about what to do about it, that happens here. Whereas PNS is carrying things up and back down, right? So I want to go through these one by one. Raising your hand in class. To do this, right, you actually need to send information down from your brain to your muscle in your biceps, let's say, to contract and put your hand up. This is going to a skeletal muscle, right? You're actually going out over here. Let me show. So you would go down from the brain and you'd go down the efferent path through the somatic nervous system to your biceps, which is a skeletal muscle under voluntary control. This would be an example where you need the peripheral nervous system, right? You actually will go down the somatic nervous system. You're contracting, right? This is an efferent response, right? So you will be in the PNS for this. Thinking about the answer to this problem. If right now, right, pretend, this could be debated. To think about a problem, you actually need to hear it, you need to see it, and that is the peripheral nervous system. But pretend all that is done and the info is just in your head right now. To think about it, right, about this problem, you're not needing more visual info or hearing. You're not needing to move, right? So no afferent, no efferent. It's really just the CNS that's working here. I believe this is the correct answer. If you weren't sure, keep going through them, right? Do process of elimination. The next one says clicking the correct answer to this problem. To go and click the correct answer, you need to take your hands and move your cursor to the answer and click. That's a somatic nervous system. You need the efferent system, right? You actually need to go and do a motor command. You need to use your muscles to do it. That's still part of this efferent pathway over here, right? Feeling the warmth up from a cup of coffee, right? That will be processed here. This is coffee. I love it. Amazing. But to get to the brain, right? Feeling the warmth of a cup of coffee, sensory receptors that are not in your eyeball this time, but that are in your fingers, right, will feel it and send it up to the brain. So it's these neurons that are going up, probably over here from the somatic sensory receptors, up the afferent division to the brain and the spinal cord, right? So that is the afferent. This over here would be the afferent system, coming from somatic receptors, actually, that feel things on the outside of your skin. Somatic receptors. Okay. And so this would not be one of the answers, because this is the things going up. That's still part of the PNS, right? The CNS is part of all of these, right? But the ones that don't have the PNS, the one that makes the most sense to me is thinking about the answer to this problem. Again, you could argue with me, well, I had to look at the problem. That's info coming in. I had to hear maybe the problem through my ears. That's PNS coming into the brain. All of those things, you're right. Then in that case, you could argue that I'm wrong, right? And this would be wrong as well. But if it's purely the thinking about the answer to this problem, this answer would make the most sense. All right. So now we go on to the cells of the nervous system. Nervous tissue, if you recall from our tissues way back when we looked at tissues, nervous tissue is made of two types of cells. You have neurons that are the basic functional units of the nervous system. They will transmit electrical impulses that we call action potentials to communicate between neurons and other cell types and to coordinate bodily activities. All of these neurons here, bzz, 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 you see 3 neurons here, they are all neurons. They are sending electrical information up and down. All of these things that we're looking at here, the little spaghettis, all the brain and spinal cord, that's pretty much neurons. They're the things that are sending the info up. When you feel pressure here, a pressure neuron will send that signal up to tell your brain that it felt that. And they kind of link together. This neuron will feel things over here, send a wire down to the next neuron, which will then send an information to the next. You see electrical signals traveling across. I want you to think of neurons as the hydroelectric wires that you see on the street, right? Remember, if you look on a street and you see a pole with the wire that goes to the next pole, and then from that pole to the next pole, I want you to think of each of those wires connecting pole to pole as one neuron. And they go one neuron to the next neuron to the next neuron, right? It's kind of like that. It literally is that because it's like electricity, right? The electrical conduction in the wires of hydroelectric poles, right? That's the same stuff that travels, that's electricity traveling down the axon of a neuron. So that's in nervous tissue. Most of the stuff that we'll talk about is these neurons here. But there are helpers. This is the main function of the nervous system, sending information and communicating. But there are helpers to support the communication, right? There are poles holding up the wires. There are little elements on the wires to help it. Insulation around the wires, right? It's not actually the rubber around the wires that conducts, it's the inside. So these are neuroglial cells. Neuroglia or glial cells are non-conducting. They don't send electricity. And instead, they support, they insulate, and they protect neurons. Okay? We're going to see Schwann cells help to insulate the axon here. We're going to see the oligodendrocytes do the same thing. Microglia help to support the neuron, right? You see this yellow neuron here? All the other cells, right? They are neuroglial cells, ependymal cells, astrocytes, microglia. All of these other cells, apart from the neuron, are called neuroglia, and they just help to enhance and make this electrical signaling work. So, again, we're going to put this on the back burner, the neuroglial cells, and we'll focus on neurons for now. What are the parts of a neuron? Okay, so as I said, the whole point of an electrical wire outside is you have one pole and you send a wire to a more distant space to the next pole. That's what a neuron does. It receives information over here at its dendrites, okay? These dendrites are projections off the cell body that receive signals. So this is the cell body, right? If I cut out everything over here and just showed you this, you'd tell me, oh, that's a normal cell. But if you look at this entire neuron, you're like, what the hell is this? That's a modified cell, right? Like the typical cell I showed you in one of the first lectures, not all cells look like that. This cell looks weird, right? So over here, you have this cell body that looks a little funky, and it has all these additions to it, right? Well, it is still a cell, it's just different. So this is my cell body. It contains the nucleus and most organelles, like the ER, mitochondria. And these dendrites, they're projections off of the cell body, right, that receive signals. So maybe these little dendrites will be in your skin and they will, let's say I push over here, these little dendrites might feel touch, right? They might be affected by the touch as I press on your skin, and bzzzt, they will signal over here to the cell body, this area, that there's a signal that came in, right? So I like to think of them as the little ears that pick up all the information, the little satellites. They receive signals. That's their job. They receive signals, they send it down the cell body to the axon hillock. A hillock is like a lock on a canal, is where at a junction, right? That's where the gates are. It's actually a name for a gate, right? This is the gateway to, this isn't a dendrite. This is a very, very long type of process. These are shorter little dendrites that are nearer to the cell body. They pick up information. This is like the transmission line that will send the info to the next hydroelectric pole, right? So over here, this is called the axon. It is the projection off the cell body, the longest one, that transmits incoming messages away from the cell body. So bzzzt, it sends it away from the cell body, bzzzt, right? So this over here is called the axon hillock. It's the junction between the cell body and the axon. And this is the axon. It's the projection off of the cell body that transmits incoming messages away from the cell. Why do we care so much about the axon hillock? Because it's the region where we decide if a signal is sent or not. If only a few dendrites send the message to the axon hillock, nothing's going to happen. The info isn't going to be sent down. For example, if something brushes you very lightly such that you don't feel it, that touch won't go in, right? If there's a big rub, then more of these dendrites will send the signal to the axon hillock that they sense something, and that signal will be sent down the axon, okay? So the axon is the projection off the cell body, and it transmits incoming messages away from the cell body to the next, to the end over here, where it's going to talk to another buddy to transmit the message to, okay? I want you to note here that around this purple axon are these little sausage rolls over here, right? These are separate cells. This is actually called the myelin sheath. It insulates the axon to speed up electrical signals. This over here, these are called either Schwann cells or oligodendrocytes. Remember the neuroglial cells? Do you see these Schwann cells that wrap around the axon here? They insulate right, the axon to speed up electrical signals. They are not a part of the neuron, actually. They're a separate cell. They're glial cells, neuroglia, and they will insulate the axon. So I want you to think of them like on the wires that are in the street for our electricity. Inside is the copper and whatever metal is making up the wire. Around it, you have this rubber to insulate it so that electricity doesn't go away. If a tree falls on it, you see sparks, right? And it short circuits. The insulation is actually useful to speed up the conduction along this wire. So it's important that you know that these are there for speeding up and making this signal a lot faster. And interestingly, you see these nodes of Ranvier, they're called. I also call them gaps in the myelin sheath. There's regions where the myelin sheath are not present. It's not like our hydroelectric wires that are all full of rubber. There's actually little gaps, right? And these gaps are there to allow the electrical signal to jump from one node to the next node. It helps to, again, speed up the electrical activity. Actually, our wires for electricity are kind of like this. They go from one pole to the next, right? So there's a little place where there's a break, and then it goes to the next one. So it's actually the same kind of concept here. There's these little spaces, these nodes, and whoever Monsieur Ranvier was, who has a big ego, called it after him. I just call them gaps in the myelin sheath. There's these spaces in between the actual myelin sheath, okay? And lastly, so here the dendrites, they pick up a pressure. They send it over here. They tell the axon hillock. If it's strong enough, it goes down the axon all the way to the axon terminals, right? The terminals of the axon, which are little buttons at the end, right? Some people call them synaptic terminals, so the ends of the synapse. And at these synaptic terminals, they release what we call neurotransmitters to talk to the next cells. So these little buttons over here are full of neurotransmitter, and they release it onto the next cell, maybe another neuron, maybe a muscle, right? It depends what exactly it is. So neurons send electrical impulses in one direction, right? They receive information at their dendrites, right? So maybe over here, maybe over here, maybe over here. They pick up the input from a stimulus, and signals travel. Once this information is sent, let's say over here, it travels to the axon hillock. Information over here travels to the axon hillock, where they get summed up and integrated, right? So, oh, I just felt something at this part of the dendrite. I just felt something at this part of the dendrite, right? And then decisions are made whether the signal will proceed down the axon or not. I always think of like if you're working in a kind of a hierarchy, right? When there's ink missing in the printer, you don't go to the CEO of the company to tell them that there's no ink, right? But so if there's just ink missing, that wouldn't allow a signal to go to the CEO. You actually need the ink to be missing, half of the company going on strike. Then over here you have, part of the department is mad at something else. And then over here you have a fire going on in one of the buildings, right? All of that would escalate to then go to the axon hillock to send a signal to the CEO. It's a bad example. I'm sorry. But you get my point, right? So there's this summation and integration and a decision that's made at the axon hillock if the signal is strong enough to send it down the axon or not, right? Is this serious enough to send it to the next neuron, right? Is there a high enough signal for me to escalate this to the next neuron? So the axon conducts this electrical signal down, and we often say it conducts and it propagates, right? Conducting electricity, propagating. You could think of the word like during the pandemic, you propagate a virus, you propagate it down to other people. Well, here you propagate it down the length of the axon, and the electrical signal goes all the way down to the end, right? The signal then arrives to the axon terminals, the synaptic terminals, and they are transmitted onto the next neuron or the effector. Here there's another neuron, right? But sometimes it's an effector. Sometimes it's like a muscle that's over here. And it will then go and maybe, the dendrites over here will then pick up the signal and do something about that. One thing to note, here I write neurons send electrical impulses in one direction. They start at the dendrites and they go down the axon this way to the terminals. This is a one-way street. Information never starts at the terminals and goes backwards. That does not happen, okay? This is a one-way street. We go this way to send information. Interestingly, neurons could be classified by their structure. Multipolar neurons, right, means that multiple dendrites are attached to the cell body. So over here, do you see that on the cell body, there is 1, 2, 3, 4, 5 dendrites here. There's multiple dendrites on here. This is a multipolar neuron, right? Where are these located? These are often interneurons in the CNS. So do you see this interneuron over here? This is found in the CNS, so it could be in the spinal cord or the brain. And motor neurons, the ones that go down, let me just remind you briefly, in different neurons, right? We have sensory neurons, we have interneurons, we have motor neurons, right? So over here, we're saying that these multipolar neurons, they're the most common. They're the ones that will mostly draw and you'll see. So interneurons, anything that's in the spinal cord and the brain, right, are going to be multipolar. And motor neurons, cell bodies in the CNS, like usually when a neuron goes to a muscle, it puts its cell body, this part, in the CNS, and it sends out its axon all the way out, and its terminals are found in the PNS, okay? Unipolar neurons are a single projection that leave the cell body. The cell body is set to the side of the axon. So unipolar neurons, here's the cell body, and you have one little projection that's coming off of the cell body. And the cell body is really to the side of the axon, right? And you see over here that you have some dendrites on this end, you have terminals on this end, and this is kind of in the middle, right, of this whole neuron. So when do we see those? These are found only in sensory neurons. So let's say over here in the skin, I have the dendrites that are over here in the skin. They send over here their impulse up, right? And they pass by this way into here. You have all of these cell bodies that are accumulated in what we call a ganglia. We'll talk about that later, but the cell bodies all collect in the ganglia next to the spinal cord. And so that's where the cell body is. And we keep going and we go to the axon terminals that are over here in the CNS. So these are unipolar neurons because they have one little pole coming out. And I don't like to just memorize. I like to understand why it's like this, right? In a cell, first of all, let's go back to our other lectures. What did we say about neurons and their ability to undergo cell division? Remember that neurons don't really divide. In adulthood, neurons don't divide, and if they get destroyed, they die. There's no bringing any neuron back. Neurons, their terminals can move and synapse and reconfigure. They're what we call plastic, but they don't really divide, right? There's no new neurons that are going to be made. So, this being said, how do we kill a cell, right? We kill a cell by attacking the area where there's the important information, where there's all of this genetic info, the DNA, the organelles. Where do we find that? We find that over here in the cell body. Look at where the cell body is for these multipolar neurons. The interneurons and motor neurons, their cell bodies are in the CNS, right? They're protected. Our spinal cord's protected in the vertebrae. Our brain is protected by our skull. So these are very well protected. Over here for unipolar neurons, let's say I had the cell body in the skin and you accidentally cut your skin, then you would be cutting the cell body, and that would be bad because that neuron would die and it would never regenerate, right? Instead, what happens is these neurons they go and put their axon terminals over here in the skin, and they hide their cell bodies further away near the spinal cord. Actually, it's right next to the spinal cord, so they're closer to the midline. They're much less likely to get damaged, and they're more protected at this area. So that's why sensory neurons, actually, it doesn't make sense to have multipolar neurons when they're sensory neurons, because they'd have their cell body over here in the skin, and that wouldn't be good, because if we cut ourselves, right, we cut vegetables and sometimes we cut our skin and we start bleeding, we'd be damaging neurons and we'd stop being able to sense things in that area, right? So this is actually a smart system to have unipolar neurons for sensory neurons, so that we don't go and damage and kill them off forever, right? When you cut a dendrite or an axon, you can actually repair it. Actually, if you cut your finger off fully, they'll ask you to bring your finger to the hospital with you, because they could stitch back the big nerves, and the axons will eventually connect together, and the wires will piece back together, which is interesting. Yeah, so over here, multipolar neurons, unipolar neurons, and to be fair, that's pretty much what you need. So sensory neurons are unipolar, interneurons multipolar, motor neurons multipolar, and it makes sense, as I said. You could memorize it, but it actually makes sense why we have this special case for the unipolar neurons. The bipolar neurons, to be fair, I don't really care about. They're just special exceptions. A single dendrite and axon converge on the cell body. So here you have a dendrite converging. You have this, then you have the axon on the other side. You see that this one cell body has the process on each end. It's not like one little blip coming off here. This little cell body here hides in what we call the ganglia here near the spinal cord, so it's protected. Whereas here, this is just really connected on each end. As I said, I don't care that much about them because they're rare. They're found in special sense organs. So in the eye, you find these types of neurons, in the olfactory bulb, in the ears. So we actually say sometimes in the special sense areas, like seeing, smelling, and hearing, that's where you'd find those neurons. Okay. So, next one. Neurons could be classified by their function. So sensory neurons, basically the afferent neurons, right? The input, right? They receive stimuli internally or externally. So inside the body, like in your stomach, or how stretches your stomach, from your heart, from your blood pressure, right? Or external from your skin, sensing touch, pain. So they receive stimuli from the PNS, inside or outside, via receptors on their dendrites. So on their dendrites, these sensory neurons, they have little receptors here that pick up either touch, pain, temperature, light, heat, sound, right? Depending on where they are, they can have different receptors. But all these sensory neurons, right, they have their receptors on their dendrites, and then they send their little process over here. They have their cell body located in this ganglia, right? And they have their axon terminals in the CNS. So the output, right, the end of this neuron, carry signals towards the CNS to send information to the brain or to the spinal cord. Here we're in the spinal cord, right? Structural types, these are called unipolar. We just saw that right now, right? And their cell bodies are clustered in the PNS just outside of the spinal cord, right? Just outside. This is still the PNS. Once we enter the spinal cord, we're in the CNS. Then you have these interneurons. They're actually the most abundant type of neurons. There could be one sensory neuron and then many interneurons that integrate and think and analyze the sensory inputs and then decide the motor plan to control an output, right? Their location, they're found only in the CNS to connect neurons together, either in the spinal cord or the brain. And their structural type, they're multipolar. They have this shape. Lastly, motor neurons. These are efferent neurons. They receive input from interneurons. So these interneurons send input to these over here. They carry signals away from the CNS to the motor effectors. So you see that their cell bodies are here. It makes sense again. You want to have the cell bodies protected in the spinal cord. This is perfect. And they send their process out, away from the effector, and then they'll synapse onto their effector. It could be a skeletal muscle, smooth muscle, cardiac muscle, or a gland. And the structural type, right, again, this is a multipolar neuron because it has one cell body with multiple little processes, right? All right. So again, let me just go, here you see the sensory neurons dendrites. You see the sensory neurons axon. You see the sensory neurons cell body, and you see the sensory neurons terminals. Here you see the interneurons dendrites, the interneurons cell body, the interneurons axon, the interneurons terminals, the motor neurons dendrites, the motor neurons cell body, the motor neurons axon, the motor neurons terminals, right? So I think I've talked about this already, right? But this is just going over what we saw already. Neurons, in terms of the sensory neurons, they start in the PNS. Let's say the skin here, they have their dendrites. They send their impulse up. They put their cell body in the ganglia here, just outside the spinal cord. And then they put their terminals in the CNS, maybe the spinal cord, or it could go up to the brain even. Interneurons, right? So then you have something from the brain come down, a signal come down, and then it synapses onto, let's say, this interneuron here, which will then communicate with the motor neuron, which sends its impulse out to the target, onto effectors. Motor neurons can synapse onto skeletal muscle if it's part of the somatic nervous system. If it's part of the autonomic nervous system that you're not in control in, then it's one of these three. The smooth muscle, like the ones around the intestine, let's say. Cardiac muscle, squeezing the heart, or glands, like the sweat glands. Alrighty, now going on to transmitting information at the synapse, we're going to see what happens at the ends of those neurons, right? So neurons, they talk to each other and to their effector across what we call a synapse. We probably talked about this back in the muscle module. And so now we're just going to recall what we said, and this should be easy. I want to define what a synapse is, though, right now. It's the location where two different neurons communicate with each other. So one neuron talking to the next. It's kind of like the hydropole, right? Where one wire comes and talks to the next wire. So it's where they meet together, right? And they talk. And so you see over here, these little, they butt out these little neurotransmitters and bind to receptors on the other neuron. That's what we're going to see, is this how they talk to each other. What is the synapse made of? The synapse is made up of two membranes and a space. So here's your presynaptic neuron, the neuron before the synapse. Here's the synapse, right? And here's the postsynaptic neuron. It's the neuron that's receiving the info. Remember, neurons only travel information in one direction. Electricity travels in one direction, bzzzt, in one direction. So here the presynaptic neuron talks to the postsynaptic neuron, and really it's just from here they transmit this way, and then this neuron transmits down information. So the membrane at the axon terminal is called the presynaptic membrane. The one that's over here on this side, that is the end of the neuron, right? And it contains these vesicles. These are called vesicles, little, I like to call them vehicles, right? Little, you could fit a few people in your car, right? The little vesicles. And they are filled with neurotransmitters. So over here you see little neurotransmitters inside. I would like, and there's different types of neurotransmitter, and different neurons are called different things depending on what kind of neurotransmitter they have. You can have dopamine neurons, serotonin neurons. There are different types. So here, adrenaline is sometimes the neurotransmitter, and that's for the fight or flight neurotransmitter. Adrenaline to get your heart pumping when you're in a stress scenario. This is really part of the sympathetic nervous system. So adrenaline, another name for it is actually epinephrine. Noradrenaline is for concentration. So this is norepinephrine. Dopamine is the pleasure neurotransmitter. This is simplifying things a lot, but yes. Serotonin often controls mood. GABA is the calming neurotransmitter. And it stops signals. It's more of this inhibition. Acetylcholine is learning in the brain, but we saw acetylcholine a lot in the muscular system. All skeletal muscles receive neurons that release acetylcholine onto them to tell them to contract, right? So this is one of the neurotransmitters as well we care about. Glutamate, you could say it's involved in memory, and endorphins have this euphoria effect. They say after running, there's a lot of these endorphins released that give this happy, I've never felt it, I guess, but this happy feeling after you run. So all this to say that the presynaptic neuron, right, here it's all electrical, bzz, 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 electricity, charges move, but then at the ends here you have these vesicles filled with chemicals that are called neurotransmitters. And these neurotransmitters, if once the electrical signal bzz, bzz, gets sent to the end, it tells these vesicles to fuse with the membrane and release the neurotransmitter in the space between the two separate neurons, and that's called the synaptic cleft. So this is where neurotransmitters are released. And then the neurotransmitters will bind onto the dendrite of the next neuron. This could be a neuron, or it could be actually a muscle or gland. Like we saw in the muscular system, this was the sarcolemma over here, and it was the last neuron in line, the motor neuron talking to a muscle. But if this is, let's say, a sensory neuron or an interneuron, it will talk to another neuron before, right? So this could be a neuron as well. And so this is called the postsynaptic membrane. It's the neuron, the membrane that's after the synapse, right? That's receiving the info. And it has these little channels or receptors that bind the neurotransmitter and open up gates to allow an electrical impulse to come in. So neurons talk to each other and to effectors across synapses, right? Sensory neurons, here's a sensory neuron with its receptors in the skin. It will send its information up to the terminals here, right? And it will talk to this postsynaptic neuron. So this sensory neuron is actually called the presynaptic neuron. It will release neurotransmitter in between at the ends here. It will release neurotransmitter out of its axon terminals, and it will release it onto the yellow, into the synaptic cleft. So from the presynaptic neuron, green, into the synaptic cleft, there's a micro space here that you don't even see between the green and the yellow. And it will bind onto the yellow postsynaptic neuron here, right, which is an interneuron. This interneuron then passes their signal to the next neuron over here, which is the blue motor neuron, right? So it will go bzz, bzz, send a signal down to the axon terminals, and here these yellow ends are called the presynaptic neuron that release it onto into the synaptic cleft, the space, and then onto the blue postsynaptic neuron, right? I want you to notice that this yellow neuron, for instance, over here it was considered the postsynaptic neuron because info was going from the green onto the yellow, so it was on the other side of this synapse. But when we're talking about this synapse here, the space between these, the yellow neuron all of a sudden is called the presynaptic neuron because it received info here, sent it down, and now it's going to transmit it. So now it's the presynaptic neuron going on to the postsynaptic neuron, okay? So, that's our situation over here. The motor neurons, they pass their signals then, here's my motor neuron, down their axon to either some kind of effector, okay? So I want you to notice here, for sensory neurons, this is from a neuron and definitely onto another neuron. There's no way that this could synapse onto another muscle or something else because the sensory neurons pick up stuff and they need to send it to the central nervous system. This is always neuron to neuron. The interneurons, they pass from one neuron to another, to another, to another, and then eventually onto another neuron called a motor neuron. So that's still neuron to neuron. But then all motor neurons, right, they pass their information from a neuron, this blue neuron, to an effector. Because motor neurons, by definition, they go to an effector to do something. Sometimes we call it the neuroglandular junction, or sometimes we call it the neuromuscular junction, the NMJ. You maybe remember this. So in this case, this is an NMJ, neuromuscular junction, because it goes from the presynaptic neuron into the synaptic cleft, the little space between, and onto the muscle over here. Sometimes this isn't a muscle, and sometimes it's a gland, actually. So this will be a neuroglandular junction. It'll go from the presynaptic neuron into the synaptic cleft onto a gland to tell it to squeeze out its substance. So that is a possibility as well. One thing to note is how neurons talk to each other across a synapse. Over here, right, in this presynaptic neuron, an electrical signal propagates down the presynaptic axon. I want you to understand that inside a neuron, it's an electrical signal. It is charges like ions, like sodium plus, that are moving around and causing electrical changes. So that's called an electrical signal. Once it gets to the axon terminals, that electrical signal tells these vesicles full of chemicals to fuse out into the synaptic space. So a chemical neurotransmitter signal is released into the synaptic cleft through vesicles. This is not electrical. This is not an electrical signal here. The electrical signal was in the neuron, right? It told these little vesicles to release. This is a chemical. It's not charged. It's not voltage. It's not electricity. And those little chemicals bind onto these receptors, right? And that will cause, these receptors will cause ions like sodium, electrical ions, to flow in and cause a new electrical signal in the postsynaptic cell. So I like to think that neurons talk to inside themselves, they talk in electrical signals, but when they talk amongst each other, they talk in chemical neurotransmitter signals, right? So the electricity is really inside the neuron, whereas the chemicals are found in this synapse area. Neurons talk to each other across these synapses, right? So I want to go over the steps again that we looked at. We actually looked at them in our muscle lecture. So electrical signals travel down the axon and arrive to the axon terminal. Calcium channels, remember that electrical depolarization, right, causes calcium channels in the membrane of the axon terminal to open. This causes high calcium from the outside of the neuron to move into the neuron where calcium is low. This calcium, remember, we don't need to know how, but this calcium is important for moving things around. And so this calcium will allow these vesicles full of neurotransmitter to exocytose and merge and blib off on the end over here, right? This will cause binding of, this is released into the synapse, right? And this will cause binding of neurotransmitters to its corresponding receptors. Do you see this neurotransmitter binding onto this receptor ion channel in the membrane of the postsynaptic neuron, or maybe the muscle, right? This binding, you see this binding causes the ion channel to open up, and it causes ions. Do you see these plus charges here? Usually it's sodium or potassium. So sodium, these ions will trickle through from high to low into the postsynaptic neuron. And if I put a bunch of plus charges inside here, all of a sudden I change the voltage, the electricity inside, and I cause a stimulus, right? I want you to note something. One thing that people get wrong often, they think that this neurotransmitter does not enter the postsynaptic cells. Some people think that this neurotransmitter will be released and will enter into this side. So let's say this acetylcholine we saw in the muscles, or let's say dopamine, right, or serotonin, this gets released in the synaptic space, but it does not enter into this side. What enters into this side is other molecules, these plus charges, usually like ions like sodium, enter in. These low neurotransmitters just bind onto these receptors to open them up, to allow charges to move in. So that's why I keep saying over here that, I said that, where is it? That really we have this electrical signal, a chemical signal, and then electrical again. Here, right, same situation, we have electrical signals, a chemical signal here, right, and then chemical released, and then over here, this is going to be electrical again because the neurotransmitter doesn't enter onto this postsynaptic cell. It just binds to the receptor to open it to allow electrical activity to enter, plus charges to enter and change the voltage. So electrical, chemical, electrical. Neurotransmitters are removed from the synaptic cleft in 3 ways. So as neurotransmitters are removed from the synapse, the postsynaptic cell could be ready to respond to another event. If the synapse stays in here, then they'll keep binding onto these receptors and keep stimulating the neuron, and it'll keep stimulating. Somehow we need to get rid of this neurotransmitter. And we can do it in 3 ways. There is something called reuptake. Neurotransmitters could be reabsorbed and recycled in the presynaptic neuron by reuptake transporters. So here the neurotransmitter got released and it bound onto its receptor. It caused it to open, caused depolarization. But then eventually it unbinds and it gets reuptaken back and repackaged here. It's a type of recycling. It's reusing this, right? Secondly, B, there's diffusion. Neurotransmitters, this space is tiny. And you know that molecules bounce around, right? So they bounce, they bind onto the receptors, they fall off, they rebind, they come off, and then eventually they might diffuse away out of the synapse and not be able to bind anymore, right? So neurotransmitters could just go away and get diffused away from this very high concentrated area and stop binding. And lastly, degradation. Sometimes neurotransmitters, they bind, they unbind, and then they could get degraded. Over here you see this enzyme that breaks down the neurotransmitter so that it can't bind anymore. This is like acetylcholinesterase that we saw in the muscular system that would degrade acetylcholine so that it didn't work. And oftentimes they'll be recycled up in the neuron. All right, so now we're going to talk a bit more about the effect of these neurotransmitters on the postsynaptic membrane of the next neuron, right? So we're going to talk about neurotransmitters, and what's to know about these is, as I said, there's many different types of neurotransmitters. some of them are termed excitatory and some are termed inhibitory. So I want to talk about what that means. An excitatory neurotransmitter means that when it binds to channels, right, it will be letting in positive ions. So over here, right, you see that this is kind of like on, let's pretend that these are the channels over here, right? And the neurotransmitters are binding onto these channels, right? So here's the yellow neurotransmitter binding onto this channel over here. If this is an excitatory neurotransmitter, it will open this channel, which allows for positive ions, charges like sodium or potassium, let's say, right? In this case, it's usually sodium. It allows positive charges to enter. And so positive ions enter the neuron, the postsynaptic neuron, and they accumulate so that the postsynaptic membrane potential will rise toward activation. So, what is to say about this? Normally, neurons are at this level of minus 70 millivolts, okay? They're at this negative voltage that we'll talk about in a minute. This is called the membrane potential. Neurons are usually at this resting minus 70. When positive charges flood in, right? When positive things come into the neuron, you see that that voltage becomes more positive, right? It goes up. The voltage becomes more positive, away from the negative side over here, up towards zero, right? You see that the voltage is going up. So an excitatory neurotransmitter, right, allows for positive ions to enter, thus increasing the membrane's potential up. This we'll see later on, but this makes it more likely for this neuron to fire and to actually send a signal. This is excitatory. This is the threshold that the neuron needs to get to. This minus 55, this is the threshold it needs to get to actually itself decide to send a signal to the next neuron down the axon. So this is exciting for the neuron if there's plus charges coming in because it gets closer to this threshold level. And this promotes electrical signaling. It promotes sending signals. Inhibitory neurotransmitter will bind to the channels, right? Here, there's these little brown triangles that bind. These are inhibitory neurotransmitter. They will open up ion channels, but they actually let out positive ions. So in this case, they actually let out, you see that these positive little ions here, this is actually potassium in this case. These positive ions will flood out into the outside, the extracellular fluid, from inside in the cytosol to outside. So this is an inhibitory neurotransmitter causing the opening of this because positive ions will exit the neuron and accumulate so that the postsynaptic membrane potential moves away from the threshold. So if I have a bunch of positives inside my neuron that exit out into the extracellular fluid outside of the neuron, then positives leaving means that the inside becomes more negative, right? If you have positive people leaving from around you and you're just going to be surrounded by negative people, then it's more negative on the inside, right? Well, that's what happens here. Here you see that, when this opens up, right, you see that boop, there's this decrease in the potential. This more positives are leaving. Another way of thinking of it, more negatives are left inside, so it becomes more negative than it was. And this actually moves away from the activation threshold. This is the threshold we need to send a signal, okay? And actually, in this case, right, we are preventing the sending of a signal. That's why we call it inhibitory. It kind of silences the neuron to not send signals, whereas this one was excitatory. It tried to up and actually send a signal to the neuron. And as I said, this is a crazy drawing, but bear with me. There are different neurotransmitters. Acetylcholine, right, excites postsynaptic membranes. Acetylcholine we saw in the muscle bound onto the muscle, and it opened up channels allowing sodium to flood in, making it more positive and closer to the threshold, and that excited and allowed for more likely generation of an action potential. That was an excitatory, and we know this because then the muscle contracted once the action potential went through, right? Epinephrine and norepinephrine are also some excitatory neurotransmitters, dopamine as well. Serotonin and GABA, on the other hand, these are inhibitory neurotransmitters that will inhibit and stop the signaling of neurons. Um, I always put this crazy drawing, and I hate it because I can't find a drawing that's better than to express this. But what I want to express to you in this drawing from a scientific paper is that this is my postsynaptic dendrite, let's say, right? My postsynaptic neuron. And I want you to understand that many different types of neurons are synapsing onto that one neuron, right? That is receiving. So many, let me rephrase this, many different postsynaptic neurons are synapsing onto that one, sorry, I take that back. Many presynaptic neurons are synapsing onto that one postsynaptic neuron, right? Here you have a noradrenaline neuron that is making, do you see these little dots here? It says norepinephrine. It's making norepinephrine in the vesicles, and it will release norepinephrine into the synapse. But here you have a dopamine neuron that is synapsing onto this neuron, right? And you have dopamine that's in these little vesicles, and it could release its contents here. And here you have 5-hydroxy tryptamine, and this over here is actually serotonin, 5-HT. So this neuron can release its serotonin onto this postsynaptic neuron. So all this to say, sometimes you have many different neurons synapsing onto one. And you see that the postsynaptic neuron actually has all the receptors. They have norepinephrine receptors, they have dopamine receptors, they have serotonin receptors. So this neuron is ready to listen to whoever is signaling to them, whoever is firing and sending out their neurotransmitters, right? So, you know, what I want you to get from this is that each neurotransmitter, right, has its own specific receptor. And a neuron can only respond to the ligand, to that neurotransmitter, if it expresses that receptor. So over here is my postsynaptic neuron, right? And if my postsynaptic neuron has a little acetylcholine receptor, it will just fit onto the acetylcholine ligand, the acetylcholine neurotransmitter. If it has a dopamine receptor and dopamine is released, the dopamine ligand, the dopamine molecule, then it will fit onto here and it'll signal on the neuron here, right? So you could get mixed signals in neurons, some that are exciting, some that are inhibiting, right? But that's why the nervous system is complex, right? And many different signals can excite, inhibit, and modulate a little bit the response. Some common neurotransmitters. So, as I said, neurotransmitters, they're chemicals that are stored in the synaptic vesicles and they're released by the neuron at the synapse into the synaptic cleft. And the ones that I want us to know for this specific course are one of them we know already. So, motor neurons that synapse onto skeletal muscle. We know that they release the neurotransmitter acetylcholine. We saw this before in the previous module. So they release acetylcholine into the synaptic cleft, into the space, right? And that causes, when it binds to its receptor on the muscle sarcolemma, that will cause the muscles to contract, right? I know we went through many more steps, so It binds, it opens a channel, sodium comes in, it goes to the sarcoplasmic reticulum, blah, blah, blah. But in general, I want you to understand that acetylcholine being released at the synapse will downstream cause the muscles to contract, right? So this is a type of excitatory neurotransmitter. Some neurons in the CNS, in the brain or spinal cord, right, they release the neurotransmitter serotonin, right? If I look at serotonin, that's an inhibitory neurotransmitter, onto other neurons that are responsible for mood, for happiness, focus. So they act as your body's natural mood stabilizer. Um, you don't need to know this, but just for fun, you maybe have heard of SSRIs. This is a type of medicine, a pill that's given for depression. They're called serotonin reuptake inhibitors, SSRI. Um, and these meds, right, they influence the neurotransmitter serotonin. They actually influence their reuptake. They will go in and make sure serotonin can stay around in the cleft and help to promote and stabilize mood during depressive episodes. So, now what do I need to tell you about? We know that there's some neurotransmitters that are excitatory, some of them that are inhibitory, right? And so I want you to recall that one postsynaptic neuron receives synapses from many presynaptic cells, right? Here I have my yellow neuron, that is the postsynaptic neuron, and it is receiving, in this case, from 3 different neurons. Do you see E1, E2, and I, right? The reason why they call it E1, E2, and I is because E1 is excitatory, E2 is a second excitatory neuron, and I is an inhibitory neuron. And so I want to give you an example here where this neuron, this yellow one, can decide to fire or not based on the pattern of electrical activity that occurs, right? Over here is the membrane potential. It's at minus 70, the resting membrane potential. And all of a sudden, E1 decides to fire, bzz, bzz, right? And it does a little bump over here. It decides to fire. And remember, this is excitatory. So this will release neurotransmitters that are excitatory, which will open channels that allow some positive charges to go in. That's why you see that the blip is going upwards and then back down, right? So you see that when E1 fires once, bzz, it goes up and then back down, right? And then over here at this time, bzz, if E1 fires again, it goes up and then back down. But do you see how we didn't reach our threshold yet? This means that we're not going to be, this yellow neuron will not be firing an action potential because it has not reached the threshold. Over here, though, if we synchronize the firing of E1, right? E1 fires, bzz, and then E1 fires soon after before it goes down, bzz, again. We have reached our threshold, and this neuron over here at the axon hillock, it will decide, oh, I have reached a high enough threshold. I can send an action potential down the entire axon to the next axon, right? So this is this over here is called temporal summation because this neuron fired in time. It fired at the right time, very close in synchrony, so that the two signals could sum up together. So temporal summation, the timing was summed up and it allowed for this neuron to fire. Now over here you see E1 and E2 at this point, they fire together, right? Bzzt and bzzt at the same time. These are different neurons, right? But they fire at the same time, and that will cause a little bit of a bump from one of them, a little bit of a bump from the other, and they will summate together, and they will reach that threshold. And so then this neuron will, at the axon hillock, will reach the threshold over here, right? And it will send the signal down, right? This over here is called spatial summation because this neuron is spatially in a different space than this one, but they summated together because they sent their action potentials at the same time. The two different spatial neurons, they're in different spaces on the neuron, right? But they summate together and they allow for you to reach the threshold over here, right? And then over here, you see a bunch of scenarios where it will not summate together. So here's E1, right? Here's I, where it goes down, right? Here's E1 plus the I inhibitory one, and you see that they fight together, right? Because the excitatory neuron number 1 would make a blip, but the inhibitory one would make a downwards blip. So they kind of just neutral out in the middle over here. These would not reach our threshold. So depending on which neurons are firing when, right, that will change a lot how our postsynaptic neuron will act. So again, as I said over here, the summation, I need to just give you a few words here. EPSPs and IPSPs. So EPSPs are excitatory postsynaptic potentials. They are potentials, right? This is a little potential that occurred due to excitation. This is another potential that occurred due to excitation. This is a IPSP. It's a inhibitory postsynaptic potential. In the postsynaptic neuron, you had a potential that was inhibitory, it went downwards, whereas this one was an excitatory postsynaptic potential. It excited and it got closer to the threshold, right? So EPSPs and IPSPs, they determine whether neurons will send their signals or not. And the combination of EPSPs and IPSPs, they could prevent a cell from reaching its threshold. So let me show you over here. Over here, this is over time, right? So this is measuring, let's say, this yellow neuron, right? Its potential at the axon hillock. So here, I'm at minus 70, right? And then some depolarization of a stimulus is applied. And this neuron has a little EPSP, right? An excitatory postsynaptic potential because it went closer to the threshold, which is somewhere up here. This we call a depolarization. Then over here, another neuron, maybe this I neuron over here, right, fires, and you have this lower hyperpolarization that occurs, right? And so this is a separate neuron that allows for the potential to go lower. And then the stimulus is removed, so it goes back to normal, back to minus 70. If I apply the stimulus of this neuron at the same time as this neuron, right, you see that this depolarizing stimulus is applied here, whereas this hyperpolarizing stimulus is applied over here. The two of them, they counterbalance, right? This one caused a bump upwards, this one caused a bump downwards, so the actual potential you're going to measure is going to be flat, right? Summation is the idea that signals add up to move the voltage towards or away from an activation threshold. Here they summated together, right? If I have a positive 10, let's say this is positive 5 millivolts. Do you see how we went from minus 70 to like minus 65? This is positive 5 millivolts. Let's actually write it down. Positive 5 millivolts, right? That this caused close to the activation threshold maybe is over here, right? This one over here, when this neuron fired onto the postsynaptic neuron, it caused to the postsynaptic neuron maybe at minus 5 millivolts, right? Well, if they fire together, these two neurons fire at the same time, kind of like over here, E1 plus I, right? When E1 and I fire together, well, plus 5 millivolts minus 5 will give me 0, right? So you see that the potential stays flat over here because they counterbalance. So it's really a game of summation and trying to stimulate or not this yellow postsynaptic neuron here. So I like this picture a lot because here at the whole point of this is to reach what we call your threshold of activation. This is your threshold of activation. The game is to try to reach this to be able to spike and send your signal, right? So here you see that, oh, one EPSP came in, oh, two EPSPs come in. I'm almost close to this threshold, but then, boom, another neuron fires and it's an IPSP. It goes back down. And then an EPSP comes in, and then an IPSP. And then a big EPSP comes in and I reach threshold, and this over here is where I reach the threshold and I send my signal. That will go down the neuron all the way to the end, right? Until this point here, no signal went down the axon, right? During this whole process, it was just a bunch of signals from the dendrite going to the axon hillock and summating together. But remember, the axon hillock is the place where we decide whether or not we are sending a signal down the axon, right? So this is the place where summation occurs. So I just want to talk about a few types of neurotransmitters that we care about. So medication and drugs that cross the blood-brain barrier. The brain is protected by this barrier. We call it the blood-brain barrier because the blood and the brain do not mix. There is a barrier that is there. If things do come in, right, they need to pass this checkpoint, kind of. So medication and drugs that do cross this blood-brain barrier to affect the nervous system commonly work at the level of the synapse. So these are all drugs that can enter the brain because they have these effects on our cognitive state, right? So it makes sense that they do get to the brain. So the first one I'll talk about is selective serotonin reuptake inhibitors, or SSRIs, as I said. These prevent serotonin from being reabsorbed. These drugs, selective serotonin reuptake inhibitors, they selectively, they will basically be preventing serotonin from going back into the presynaptic neuron, and they'll keep them in the synapse. So let me show you a picture over here. Here you have, this is your serotonin receptor, serotonin, let's say, right? So this is the receptor for serotonin. Let's say that this purple molecule is the serotonin, right? Serotonin usually binds over here. It causes a little blip over here, bzzzt, right? And then it unbinds. And then sometimes it rebinds and then it unbinds. And then at some point, right, it goes up through this serotonin reuptake little channel, and it'll be recycled up here for the next time that there's a signal. What SSRIs do is they are selective serotonin reuptake inhibitors. They inhibit and they will block. These are big chunky molecules like this. They will go and clog this pore over here, and therefore serotonin cannot be taken back up into the presynaptic neuron. So serotonin actually stays in the synaptic cleft and keeps binding onto these channels here, right? And it keeps binding, and the serotonin neurons will keep having their effect, and therefore you will be more in a happier state. It'll cause this more happy state and stabilizing your mood. So that's serotonin reuptake inhibitors. We have cocaine is another drug that can cross the blood-brain barrier because it has different effects, right, on mental state. This is a drug that will inhibit the removal of dopamine from excitatory synapses. So it blocks the removal of dopamine, and this leads to feelings of euphoria and alertness that are typical for users of this substance. There's also another drug that is called MDMA, and these also that are used recreationally will be increasing the release of serotonin, norepinephrine, and dopamine from certain presynaptic neurons, and they actually prevent their reuptake. So they keep those neurotransmitters in the synaptic cleft, and this leads to symptoms of euphoria, alertness, and increased empathy. So these all play around. What I want you to see is that all three of these, right, drugs are affecting mood. Mood is controlled in the brain, in synapses, and so it makes sense that these can cross the blood-brain barrier to get into the brain and affect neurons, right? This one is used therapeutically. SSRIs are therapeutically used to help patients. They are given at a correct dose to help patients. These two drugs over here, they are used more recreationally, and there are, because they affect the dopamine system, they do have the ability to affect patterns of addiction, and that's why these are studied quite often. A few other drugs. We have benzodiazepines. These, let me talk about their mode of action. They enhance the effect of GABA. GABA is an inhibitory neurotransmitter, right? And so, what is the result of that? So, here's GABA, right, that's being made. And GABA usually binds onto these receptors over here, but benzodiazepines will block the receptors and reduce the effects of these neurons. These neurons will fire less because benzodiazepines will be inhibiting these neurons. So this will reduce neuronal excitability, and this is a calming, anxiolytic, and a sedative effect. It relaxes patients, and it's used in a clinical setting oftentimes. Amphetamines, on the other hand, their mode of action is they will increase dopamine. Amphetamines at a therapeutic dose will increase dopamine and norepinephrine, and they're stimulatory neurotransmitters. So over here you see that there's some release normally of dopamine and norepinephrine, right? And they will increase, amphetamines really increase this output of these. And so you will be more alert. You will have a little bit of euphoria, increased heart rate, and focus, right? This is often at a very low dose given to people with ADHD. It helps to with focus and alertness. It does have some side effects, as I said, of heart rate, right? But if you give a low enough dose, it's not as bad. But these amphetamines at a low dose are used therapeutically as well. And opioids over here, they are drugs that bind to the opioid receptors, and the result is they inhibit pain signaling. They cause euphoria as well, and one side effect is that they cause respiratory depression as well. So, but this is a common pain drug that's given to reduce pain. Now, we're a bit careful now with giving opioids. There's a big problem with addiction of opioids, so clinicians are extremely careful when they prescribe opioids to patients. So these are all neurotransmitters that have this modulatory effect. They're not creating a new neurotransmitter that's being released. They're changing the volume of release of certain neurotransmitters. We call these psychoactive drugs. They actually have an effect on the brain, and they do that by modulating the release. So here we're enhancing the effect of GABA. We're increasing release of GABA to, inhibit neurons. And over here, we're really increasing dopamine to heightened alertness. Alrighty, so the next topic is going to be on the spinal cord and spinal nerves. So first of all, where is the spinal cord, right? Over here you have the skull, right? A piece of the skull. This is actually the backbone, the occipital bone. And you see this hole over here, okay? And this is called the foramen magnum. Foramen means a hole. It's a foramen. Sometimes they say foramen are holes in certain bones, and magnum just means very big. This is one of the very biggest holes, right? You have little holes over here where the spinal nerves come out, but this is a humongous hole, right? Because the spinal cord is is pretty big. So this is the foramen magnum, and the spinal cord, right? It passes it's actually continuous with the brain, right? So it begins by passing through this little hole over here in the skull. That's called the foramen magnum. And it's continuous with the brain, right? It's not like the spinal cord stops and then the brain starts. The spinal cord morphs and becomes the brain. So it passes through this hole in the skull called the foramen magnum. And then down here, right, the rest of the spinal cord, you don't see the spinal cord here, okay? The spinal cord extends in what we call the vertebral canal, right? A canal is like a passageway. And so here's the canal, the empty space. So I want you to imagine we took out one of these vertebrae and we're looking at it down, right? So let's take out one of these vertebrae and look at it downwards. This little spiny process here is what you see sticking out on the sides here, right? So that's the back, and this is the front of this is kind of like this part over here, the body of the vertebrae. And the spinal cord extends through this vertebral canal, right? And it's basically protected. All of this part here, right? All these little bones together, they form what we call the vertebral column. It's a column of stacked little bricks that are all little pieces like this. There's 1 vertebrae, 2, 3, 4, right? And we actually name them in specific ways. And so they're all stacked over here, and they form, it's like one hole in the middle where this spinal cord can pass, right? I want you to imagine these as like a bunch of little donuts all stacked on top of each other. You will have one hole in the middle of all those continuous donut holes, right? So that's where the spinal cord will live. One thing about the spinal cord, right? Over here, you don't see the spinal cord. It's inside the hole, inside the vertebral canal, right? Inside over here. So you don't see it. You see it over here at the top peeking out. But inside, right, if you took off the bones, what you would notice is that along the length of the spinal cord, there are these little projections that come on or off of the spinal cord, right? Do you see these over here that are kind of coming on or off? These are the 31 pairs of spinal nerves, right? Do you see how there's one on this side and another on this side? One on this side and another on this side. So there are 31 pairs left and right of spinal nerves in total, right? There's 62 spinal nerves, one for the left, one for the right. And each of these, like this one over here, will do the same thing as this one over here, but on the opposite side of the body, right? So there are these 31 pairs of spinal nerves. Here you only see 1, 2, 3, 4 pairs. And they're leaving or entering the spinal cord. I want to use both words because it's a little bit tricky. Nerves stemming from the spinal cord, right? These are the nerves, right? The spinal nerves. They're said to be mixed, okay? And this is a bit of a difficult concept to some. If I chopped, right? So this is the spinal cord. These are the nerves coming out. This is a little diagram trying to depict this. So here we have the spinal cord, and we have these spinal nerves on each side, one for the left of the patient, one for the right. And if I chopped over here, right, the nerve, what you would see is this, right? This is one peripheral nerve. And what you need to understand is that in a nerve, right, a nerve is not the same as a neuron, right? In one peripheral nerve, there are many neurons. This is one neuron. Actually, it's the axon of one of the neurons. Maybe it started down here and it had its cell body, right? And it's sending its axon, right? So this neuron here, there are 1, 2, 3. There's so many little axons, neurons in one peripheral nerve. So if you cut this over here, you're not going to affect one neuron. You're going to affect multiple, okay? And what I need you to understand about these peripheral nerves is that they're said to be mixed because some of these neurons are what we call sensory neurons, right? They sense things. So some of these neurons will be sensing things from out over here in the finger, right? And they will send information up. Maybe, you know, all these over here are sending information up. Maybe these over here are also sending information up this way. Remember that neurons only conduct signals in one direction. So this is a very important note to make. So maybe these over here are sending information up over here and going up to the spinal cord and probably up to the brain, right? On the other hand, right, this is said to be one peripheral nerve is mixed. So they not only have sensory neurons, they also have descending motor neurons, right? So we have ascending sensory neurons going up to the CNS, up to the brain even maybe. And then we have half of these, right, that are sending information down, right? So over here, imagine that I extend this little axon here, right? This will have its terminals somewhere over here, right, on a muscle. Let's say this is a muscle over here, right? So it's going to be sending its information down from the CNS, from the brain and spinal cord, and it'll be going down this way, right, instead of going up. So when you chop a peripheral nerve, you see half the fibers are going upwards and probably the other half are going downwards, right? I don't know the exact proportion, but think of it that way. So spinal nerves, they have confusing information because half of it's going in one direction, the other half are going down, right? So I like to think of it like a highway. On a highway, you have, you know, half of the lanes that are going upwards and the other half are coming downwards. This is a bit more chaotic. We don't know which are which, but you understand the point of this, right? Some are going northbound, CNS bound to the brain. The other half are going southbound to the muscles and the effectors. All right. Where is the spinal cord? So again, I really like this picture because over here you see that this is the spinal cord, right? Up until about here, right? Do you see the tip over here where it ends, right? That's what we call all of this stuff here is the spinal cord, right? The spinal cord is part of the CNS. Spinal nerves or peripheral nerves, right, are part of the PNS, right? So these little spaghettis coming out, do you see one, two, oops, three, right? There's all of these little spaghettis that come out, right? They are part of the PNS. They're the nerves that are coming in slash out, right? These are the, over here, if I come here, you see these little spaghettis here coming out. That's the spinal nerves. So the spinal nerves, they're identified based on where they actually exit the vertebral column. So not where they originate. Why do I say that? Let me erase over here. You see that this C1 up here, right, exits out this way, right? Pretty parallel. C2 also, C3 also. As you go down, do you see how these over here are kind of veering downwards and kind of turning around? And then over here, this last orange one here is really going down and veering out pretty far down. And then this one over here, this pink one, right, goes all the way out to the bottom and veers over here. So that's why they say over here, they're identified based on where they exit the vertebral column, not where they originate on the spinal cord. Surprisingly, right, your spinal cord, right, a lot of people think it goes right to the start of your butt, right? But that's not true. The true spinal cord, right, ends about kind of halfway down your back, maybe a little bit lower, but it's really not that big. The spinal cord is not that big of a structure. You can look it up on an image in Google. You will see that the spinal cord really, relatively, it ends, as I said, like in your middle-ish back. And the rest of this, all of this stuff, are just nerves, right? These are all spinal nerves that are coming out. So now we need to know how to name these nerves, because sometimes that is quite useful. So over here we have some cervical spinal nerves. There are 8 cervical spinal nerves. Cervical means neck, remember that? So these are the 8 cervical ones. They come out in the neck region approximately. So C1 through C8, okay? Thoracic, these are the thoracic ones. There are T1 to T12 thoracic spinal nerves. These over here are in the thoracic region, right? And then you have lumbar. Remember, lumbar is like lower back. So this over here is L1 to L5. You have 5 of these lumbar spinal nerves. Then we have the sacral ones, S1 through S5. They're in the sacrum. The sacrum is a bone, this bone over here. And then coccygeal, right? The little coccyx at the end over here. That's the coccygeal one. There's one of those nerves. So that's the situation that you have for these spinal nerves. Now, what is a ganglia? A ganglia is just a term that whereby a ganglion consists of several neuron cell bodies in the peripheral nervous system. So it's a collection of cell bodies in the PNS, okay? So for example, over here, let's say I had I'm going to put it in blue actually. Let's say I had this sensory neuron coming up, right? And you know that the little dendrites over here are maybe sensing things in the skin, right? The little dendrites, and they're sending information up. Remember that sensory neurons, they accumulate their cell bodies over here in this region, right? So they're going to end up going up, but all their cell bodies are accumulated here. So here's another sensory neuron, right? It has its dendrites over here, right? And so you see that here, the little sensory neuron puts its cell body here, and it continues up. So it's a collection of cell bodies in the PNS. And you often see these ganglia over here. Each spinal nerve has its own ganglia with all sensory neurons of a certain body region. So let me go see if we can find it here. You kind of see this bump over here. That might be one. You see a little bit of a bump here. That might be one here. It's a bit hard to see in this drawing. I don't really have a better drawing. But basically, this spinal cord, right, in the dorsal part, you're going to see that there's this bump, right? And that's just a collection of those cell bodies, and we call it a ganglia. So a ganglia, as I said again, it's just a collection, concentrated collection of cell bodies, and we see them along the spinal cord. Do you see over here? You see one here, because remember I said that they collect all their cell bodies close to the spinal cord because it's kind of a little bit more protected in that area, right? So this is a good spot to have all the ganglia, all the collection of cell bodies of the sensory neurons, right? Remember, if we had multipolar neurons for sensory neurons, they would all be in the tissues themselves, like maybe in our fingers or on our skin, and they'd send their axon, but the cell bodies are very important to protect. So instead, sensory neurons, they are unipolar, and they put all their cell bodies in these ganglia. All right, so that's all I had to say about the spinal cord and spinal nerves. Now we're just going to finish off and take a look at protecting the central nervous system. So we have the first thing to look at is the meningeal layers, so the meninges. Meninges cover the brain and the spinal cord. The meninges are three specialized connective tissue layers that protect the CNS, okay? So the brain and the spinal cord, yes, the brain has the skull, right? Do you see this skull? We know that. Yes, the spinal cord has vertebrae, bones, right? But on top of that, there are these connective tissue sheets, right, that are very good for protecting. So over here is our brain, right? Do you see it go up and down and make these little curves over here, right? This is our brain. We have we have three layers. So we have the dura mater, arachnoid matter, and the pia mater. And the way to remember this is there's this little pad, right? If you remember pad, that's the letters of each of them from the innermost to the outermost. And you know that the meninges are padding the brain, right? So the innermost is P, it's the pia mater. So this over here, right? I'm going to actually go and put it in green because that's the color we use. This little tissue here is the pia mater. It's the deepest layer, and it makes contact with the actual CNS, either the brain or the spinal cord. So that's the one touching on the brain. If you dissect this, you actually see it go in all the crevices of the brain and go deep into the tissue. It's very stuck on the brain itself. Then we have over here the arachnoid matter is the middle layer. So here's my arachnoid matter, okay? And I like to think of it as arachnoid because over here, do you see these little specules? These are like columns that are holding up this wall over here and making a space over here, right? You know, like in a basement, there's columns to hold up the ceiling. Well, these are like little columns that hold up. You could actually go for a little walk through this, right? And pass in between the columns. I like this name because it sounds like arachnid, which is a spider. It looks like spider webs. These little columns that it's making with the pia matter look like spider webs that are here. So arachnoid matter, that's the middle layer. And lastly, dura matter. Dur in French, or durable in English, means tough, right? Very firm. So this over here is my dura mater. It's the outermost layer. It makes sense that, you know, like on a house, right, you have the walls and that inside, but on the outside you have this big roof, this thick roof to protect very much on the outside. So this dura matter is on the outer layer, and it's a bit thicker. and it's the most superficial. And then we hit the bone, the actual skull or the vertebrae if it's the spinal cord. Okay? Just out of curiosity, you might have heard of the term meningitis. Anything itis is like an inflammatory response, right? And meningitis is actually a condition where the meninges will get infected and will start to kind of swell up, right? So this is a disorder of the meninges. So maybe you've heard of that before. Okay, the next protection. So we saw the meningeal layers, check. The next protection is the blood-brain barrier. Sometimes they abbreviated the BBB. So the blood-brain barrier, actually there's two that we'll see, the blood-brain barrier and the blood-CSF barrier. Okay, the BCB, the blood-CSF barrier. So I just want to start with an intro. Why do we have barriers in the brain? And this is another protection of the brain. What are the cellular barriers? Barriers are generated by neuroglia, mostly astrocytes and ependymal cells. And these are some neuroglial cells that protect the nervous tissue from components in the blood. So apart from meningitis, right, you don't really hear about like having a cold or a virus or a bacterial infection in your brain. It's not impossible, but it's a little bit rare, right? You don't hear this as much, and there's a reason for that, right? Our lungs are an open passageway for microbes. There are protections, but it's not as good as the brain. And sometimes you get respiratory disorders, respiratory bacterias or viruses, right? COVID or some kind of pneumonia or just a little pharyngitis or laryngitis. So you hear about that in the resp system. You hear about different digestive bacteria that can affect your digestive system, right? But you never hear about it in the nervous system. You barely hear about it, right? And if you do, it's pretty serious. So why is that? That's because the brain and the spinal cord, the CNS, is so protected. It has what we call the blood-brain barrier and the blood-CSF barrier, where sometimes bacteria and viruses do get into your blood system and can be transported everywhere to your lungs, to your digestive system. It is a possibility, right? There are instances where bacteria get in. There are instances where viruses get in. But this blood system, which could go everywhere and kind of carry around that bacteria or virus to any organ system, does not get oftentimes to the CNS because of these barriers, right? These barriers, the blood, right? And also the CSF that we'll talk about, these barriers are completely intact in the CNS, and only with a few exceptions in the CNS, like the hypothalamus and the pineal gland. Other than that, they extend over all external surfaces and internal surfaces of the CNS. So any blood vessel that goes close to the brain and has access to neurons close to it will have this barrier. Actually, you see this, do you see over here this blood vessel that's near some neurons? You see neurons outside over here. Well, you see these little astrocytes, and we'll see ependymal cells that are just covering and making a barrier with that blood vessel. And I'm not saying that nothing gets to the brain. We do know that the brain needs stuff, right? But a lot of bad things do not get to the brain, right? And the spinal cord. So we'll talk about these two barriers together. These barriers are what we call selectively permeable. They allow certain things to pass, but not other things. What could pass through the barriers? Well, you know, right? So here's the blood vessel here, right? Do you see my little blood vessel? And so here's my blood vessel, and here are the, cells of the blood vessel. And then here's an astrocyte that puts its little foot out, right? Astrocytes look like little stars, and they put their feet out and they kind of block things from entering. But there are some things that will be able to pass. So, for example, respiratory gases, oxygen and CO2, we saw that these were non-polar. These were lipid soluble. They were able to pass through the plasma membrane of cells, right? And they could just diffuse across. So, we know that oxygen, oxygen and CO2, they can pass through and go right through. What else? Larger solutes like amino acids, glucose, ions, sodium, potassium, they actually have specific transporters, right? Do you see these transporters? That'll bring them across. Just like in a cell, a regular cell, we have certain transporters that can bring things across. So glucose, amino acids, and some other stuff, they can enter in and go because they need to go to the neuron, right? The neuron is like over here somewhere, chilling in the brain. We need these transporters to cross them because these things are water soluble things, and this is a lipid membrane here, right? So we need transporters to bring them through. What cannot pass through the barriers, right? What is it that stays out of the nervous system? Most proteins, most toxins, most drugs, most viruses, and most bacteria cannot pass freely through these barriers. This is good. So the brain is like overly protective compared to all the other organs, and really stops thanks to these little astrocyte feet that wrap around here, right? And to these tight junctions in the blood vessels. A lot of things that normally could sneak through to an organ, they will not be able to get to the neurons in the brain and the neurons in the spinal cord. However, right, a little caveat here. Some drugs do. Any psychoactive drug, anything, any drug, any medication that affects the mental state, right, of a patient will be able to pass through the blood-brain barrier. So oftentimes pharmacists talk about, different drugs that are able or are not able to pass through the blood-brain barrier. And even if a drug, even though it might not have a direct effect, right? If a drug is known to be able to pass the blood-brain barrier, that needs to be said, in the actual when promoting this specific drug, right? This needs to be said, and often you'll see these commercials of new drugs that are coming out, and they say side effects may include, and then they list out all these mental health disorders. If a drug passes the blood-brain barrier, they need to, there is a possibility that it's affecting psychoactively your brain, right? So they need to say that. So that's why they have the list of, depression, anxiety, blah, 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 that they go on and say. So even things like caffeine can pass through the blood-brain barrier. You are psychoactive. Your brain actually, your consciousness is going to be changed by caffeine, so it must pass through the blood-brain barrier, right? Anything affecting your mental state, right, is probably passing through the blood-brain barrier. But a lot of drugs, a lot of viruses, a lot of bacteria don't pass through, which is good, okay? Even when they design drugs, right, if they want to be treating a mental health disorder, they need to make sure that it passes the blood-brain barrier. There's no use making a drug that's going to bind to a neuron if it can't pass through the blood-brain barrier. So that's important. So what is this blood-brain barrier? We're going to start by the blood-brain barrier, then I'll talk about the blood-CSF barrier. The blood-brain barrier is a cellular layer between the blood, any blood vessel near neurons, and interstitial fluid of nervous tissue, right? So over here, probably you have some neurons here in the brain that are hidden. And this is a little blood vessel passing by the neurons, right? Any of these blood vessels, right, is actually going to be composed of 3 layers. So you're going to have one that's in contact with the blood. It's a continuous endothelium. Endothelium is the same thing as an epithelium, but for blood vessels. So endo inside, it's the inner layer of the blood vessels. Endothelium is often used when we talk about blood vessels. So this is in the capillary walls. So you see this thin, simple squamous epithelium. However, if you zoom in here, do you see these tight junctions? This is one cell and this is another endothelial cell. And you see these very tight junctions, right, between the 2 cell membranes. So this over here is the first part of the blood-brain barrier, that those cells have tight junctions. In other organs, it's not this tight, right? There are some, but it's a little bit more permeable. Secondly, the blood-brain barrier consists of a layer of anchoring proteins that prevent certain solutes from passing and creating a thick basement membrane. So, all epithelium lies on a basement membrane, but this basement membrane over here, right, is a special one that's thicker and doesn't allow certain things. So that's the second part of the blood-brain barrier. And the third and probably most important part of the blood-brain barrier is these astrocytes, right? This is an astrocyte with many feet, 1, 2, 3, right? And then maybe this is another astrocyte's feet, right? You saw it over here, right? That they had their feet coming down. These astrocytes, right? They wrap around their little feet around the endothelial basement membrane, and they aid to form the tight junctions, and they form this third layer of filtering out, right? So that's your astrocytes here. So what makes the blood-brain barrier special? Tight junctions occur between the endothelial cells of the capillaries to prevent the passage of most molecules between these cells, unless they can pass through the cells. Molecules are transported into the interstitial fluid. If a molecule does want to go to a neuron, right, it needs to pass through the endothelial cell, pass through the basement membrane, and pass through the little astrocyte feet, right? Maybe between them to cross over, right? So there is a lot going on here that's trying to block something from entering to the neurons, right? Over here, there'll be some neurons. So that's what the blood-brain barrier is. How do some molecules get through the blood-brain barrier? Well, some of them go through what we call simple diffusion because they're lipid soluble, because they could pass through the plasma membrane, the cell membrane. Some molecules can just simply diffuse through. So here you see lipid soluble transport. Some molecules like oxygen, CO2, or steroids, they can just go through and enter into the brain. Some molecules need active transport and facilitated diffusion, so they need little carriers to help them get through to the other side. And some molecules, so these would be some water soluble, some molecules actually need vesicular transport via endocytosis, so they actually bind to the receptors and then they bleb all together and they get pushed onto the other side, and then they could enter into the brain, right, to bind to a neuron. So this is like an intense screening process that's there, right, like at the airport, that's like blocking a lot of things from entering. So that's the blood-brain barrier. Now I'm going to talk about the blood-CSF barrier. So over here, right, protecting the central nervous system, what there is to know about CSF is that in the brain, there are these chambers, actually. We'll talk about the CNS, so the brain and spinal cord, there's these chambers and spaces that are connected to each other so that a substance that's called cerebrospinal fluid can circulate and continuously flow in a unidirectional manner. Okay, so this includes 4 ventricles of the brain. I first want to just introduce these spaces to you. Okay, so I want you to imagine them as like a mini aquarium inside the brain and spinal cord. All this pink stuff here, that's neurons, right? But inside the brain and in the spinal cord here, there is like an aquarium with liquid in it. That's called cerebro, like the cerebrum of the brain, spinal, like the spinal cord, fluid, CSF, right? It's fluid that goes in the cerebrum and the spinal cord. And as I said, it includes 4 ventricles. Ventricles, we're going to see this in the heart, but it's not the same thing. Ventricles is a space. It's a space, in this case, in the brain, right? So there's 4 of them, 1, 2, 3, 4, that's in the brain. And then there is the central canal down here, right? That's in the spinal cord. So I need you to know the names of these ventricles. So there are 2 lateral ventricles. Lateral means like in this plane, and you see that there's 2 of them. This is a side view, and this is a front view, right? So this is ventricle, the one lateral ventricle, and the other, they're lateral. They're both on each side. Or you can call them ventricles 1 and 2. So ventricle 1, ventricle 2. Right, 1 and 2. Here on the side view, you don't see them as nicely, but they kind of go like this. And they piece together. If you see in this view, these lateral ventricles, they piece together here. They have a little projection that goes into ventricle number 3. So the lateral ventricles connect down here into ventricle number 3. Okay? And ventricle 3 over here, here's another little aquarium space in ventricle 3. And then ventricle 3 is connected through a little space into ventricle 4, a bigger aquarium, right? So you have aquarium 1, 2, 3, and 4, right? I need you to think of them as 3D aquariums where fish can swim, because they are 3-dimensional, and there is fluid in them. There's no fish, but imagine so that you can see it as like an actual fluid space. And lastly, ventricle 4 is connected to the central canal over here of the spinal cord. So it goes right down the middle of the spinal cord. And maybe, let me go back to a picture of the spinal cord. Maybe you've seen in the spinal cord that in every picture of the spinal cord, if we cut it in 2, you see like this gray matter over here, right? And you see this hole in the middle. That's that central canal, okay? This is the hole that we're talking about over here, right? That's this central canal in the middle of the spinal cord. If you cut it in 2, you would see the hole from up above. So now, just to add some more annoying terms, there are connections between chambers. So ventricles 1 and 2, they actually connect to ventricle 3 through what we call the interventricular foramen. Remember, a foramen is like a passageway, a hole, right? So this hole over here, actually it's easier seen here. Ventricle 1 is here, ventricle 2 is here, and they each have a little interventricular foramen that feeds into the 3rd ventricle. Okay, between the ventricles, foramen, a little hole, a little passage. Then ventricle 3 connects to ventricle 4 via what we call this little thing here, right? So here 3 connects to 4 via this little passage, which is called the cerebral aqueduct, right? An aqueduct is like a little passageway of water that could go down an aqueduct, right? So this is where CSF could go down to ventricle 4. And so that's, now I've given you all the anatomy. So I've given you all the names of the ventricles and the little passageways. And actually, we're going to see that, CSF flows from ventricle 1 into ventricle 3, from ventricle 2 into ventricle 3, then ventricle 3 goes to ventricle 4 and down the cerebral canal. What is the function of CSF? Like, why do we have this, right? again, I really like this picture here because it makes you think. This is like sliced into the brain, and it makes you think that this is really a fluid, right? That goes around and it bathes the entire brain. So this fluid, this CSF, is just bathing these ventricles and bathing the brain, the neurons, right? So it's passing around all these regions. So the functions of the CSF, I could kind of break it up into two pieces. First of all, they are a physical protection. So this fluid bathes both the organs. So it bathes the brain and the spinal cord. You see here it's bathing the brain here. Here it's bathing the spinal cord here. And by bathing these organs, it's absorbing any trauma or shock, right? If I threw you off a building, I think you would rather land on a waterbed than land on cement, right? That's because water has this ability to absorb some of the shock, right? When you land. So fluid, right, has this ability to absorb some of that shock. It's kind of, I like to think of it like the airbag of the brain and spinal cord, right? But it's like a water bag instead of waterbed, okay? So that's one of the reasons why CSF is there. If you get concussed or whatever, then you won't damage as much. If you hit your head, right, unless it's a really hard hit and you can get a concussion, it's going to help to absorb some of the shock. I'm not saying you can like rough around this organ over here, but it is somewhat of a protective mechanism on top of the skull, on top of the meninges, right? It does help a lot. The second reason that this CSF is important is because this actual liquid here is not just water. The brain, as I told you, is bougie, and it does not directly contact the blood. Remember the blood-brain barrier? That we don't have access to a lot of things in the blood. So instead, right, instead of having all these little vessels carrying blood and being close to the neurons, what the brain likes to do is instead use CSF to distribute a lot of the nutrients, especially inside the CNS and on these outer portions, right? So instead of using primarily blood vessels to go and feed the neurons, the CNS will use CSF to distribute nutrients, gases, wastes, and hormones to the CNS. All of these neurons in here, right, and in here, they need oxygen. They need to get rid of their CO2. They need nutrients like glucose, and they need hormones, right, to contact them. So to do that, right, what's going to happen is that in certain regions, we're going to have this blood CSF barrier where the blood vessels, right, will contact a wall, right, and put all of their things that they need into this fluid, the CSF, right, and then go away, so that this CSF is what circulates in the brain and spinal cord instead of blood vessels, okay? So where does CSF contact the brain and the spinal cord? CSF bathes and exposes most of the CNS surfaces. So CSF is found in the ventricles of the brain over here, right? In these ventricles. It's found in the central canal, right? Do you see over here the spinal cord, the central canal of the spinal cord? So it bathes this, and it actually bathes, so these are inner portions of the brain and spinal cord, right? This ventricle, so here you have the lateral ventricle, one of them only. You have the interventricular foramen that goes to the third ventricle, so it's bathing all of these brain bits over here. And then you have, then we go down the cerebral aqueduct to this fourth ventricle. So we bathe this cerebellum and these regions of the brain stem. And then from the fourth ventricle, it goes down into the central canal of the spinal cord. It bathes the inside of the spinal cord. But on top of it, this is the inner parts of the brain, the inner parts of the spinal cord. On top of it Then it'll circulate on the outer surfaces of the brain and spinal cord in what we call the subarachnoid space, right? Let me go back to the meninges here. Remember the meninges, we had this pia mater that was over here. We had this arachnoid matter. Well, this space with the little specules here is called the subarachnoid space. It's below the arachnoid, right? And it's the subarachnoid space. And this is where the fluid, the CSF, will go through, okay? So, again, we're going to go through over here, this subarachnoid space. The pia mater you don't see in this picture, but you do see in pink the arachnoid matter and the gray the dura mater. All in this space here, you will have the subarachnoid space that is going to allow for the CSF to bathe. Do you see how it's touching all the outer parts here of the spinal cord, right? On this side, around it. And it'll go over here all around the brain, the outer parts of the brain. So CSF is truly covering a lot of ground here for the brain and the spinal cord. How is CSF made? Well, CSF is made in certain regions. So blood plasma passes through and is filtered in regions that are called the choroid plexus, okay? So there are regions, you're going to see them here in the purple. These are called the choroid plexus. A plexus is like a grouping, right? So these are like little groupings of junctions between the blood vessels and the ependymal cells that we'll see. So blood plasma passes through and is filtered by these choroid plexus to produce CSF. So what's going on over here? I want you to see that as blood vessels are coming to the choroid plexi, right? You see that there's the neurons are over here in the brain, but these blood vessels are covered by the astrocytes and they're protected, right? Maybe oxygen and nutrients can pass through and waste can go in, right? But not much else can pass through. So then, right, these blood vessels eventually get to this structure called the choroid plexus. And these blood vessels, right, they kind of loop over here in these choroid plexi. You see them here. Boop, 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 boop. They make these little grooves over here. And over here, right, what happens is that this is where we produce CSF and we put it into the cerebrospinal fluid. Okay, so we're actually going to make the CSF here and put glucose into here. We're going to put good amino acids, right? We're going to put all the essential things for the neurons. And eventually we can remove some wastes over here. So what is the choroid plexus? The blood cerebrospinal fluid barrier. So we had the blood-brain barrier over here, but the blood cerebrospinal fluid barrier is the barrier that occurs at the choroid plexus. It's a barrier between the blood vessels, right? All these blood vessels are going to the brain in the aim to get to this region here where they will become this blood CSF barrier. So it's this organization of capillaries over here, right, that are lined with these pink cells. Do you see these pink ependymal cells, they call them? This is a type of neuroglia. These ependymal cells, they have tight junctions. Do you see how they're tightly joined together? And these cells are specialized because these cells have these little ion pumps at their ends, and they can decide what goes into this fluid. They will decide what makes up this fluid. If they want a bit more glucose, they'll put a bit more of that, okay? So they are the filtering, and they allow for the creation of this CSF, right? So basically, where is this CSF made? The choroid plexus is found in all four brain ventricles. Here you see it in this top lateral ventricle. Here you see it in this bottom third ventricle. You know that there's two lateral ventricles, but we don't see it when it's cut like this. And then down over here, you see it in this fourth ventricle. Okay, so the choroid plexus, let me show you here. Here's the one of the lateral ventricles. You see this red little thing, and it's making, you see the arrows making CSF, right? That's where, do you see these blood vessels coming in and going to these choroid plexi, right? That's because they're making CSF there. So here's the lateral ventricle choroid plexus. Here's the third ventricle choroid plexus. And then down here in the fourth ventricle, again, you see little blood vessels arriving here. And this is the fourth ventricle's choroid plexus. It adds even more CSF. So that's where the fluid is made. That's where this CSF is made, right? These are technically the boundaries of the blood CSF barrier, okay? So the choroid plexus is found in all 4 brain ventricles. The choroid plexi are lined with ependymal cells, as I said. Here you see these ependymal cells that filter the plasma, and they create CSF to fill up each of the ventricles. I want you to think of these red choroid plexi as filling up these ventricles, and it's kind of the hose that's filling up the aquarium here, filling the aquarium here, and this one is filling the aquarium here, right? And on top of that, you know, when you fill up an aquarium and they're connected to other parts, that could move the fluid through, right? So now we're going to talk about the flow of CSF and where it goes. So let me go through the story of how CSF moves around in the CNS, okay? So step 1, the choroid plexus of ventricles 1 and 2. So this over here, you see this is the lateral ventricle, probably ventricle 1 or 2. And then on the other side over here, you don't see it, there would be the second ventricle, right? So this choroid plexus here, it makes, it will go and make some nutrient-rich CSF with full of glucose, full of oxygen, full of good things for the neurons, right? So it'll make that and put it over here into this ventricle 1 or 2, I don't know which it is. Then that CSF will flow down via the interventricular foramen here, right? So this is the interventricular foramen between ventricles 1 and or 2 and ventricles 3, right? So it'll go into ventricle 3. Over here, right, step 3, the choroid plexus, this choroid plexus here, of ventricle 3 makes and adds more nutrient-rich CSF to this region, right? So, you know, we added CSF here, and then it flowed down over here, but on top of it, this choroid plexus is adding even more CSF here, right? And filling up this tank over here. So we're filling it up even more, right? And then all of this liquid from both the up here and down here will flow down, step 4, via the cerebral aqueduct into this ventricle number 4, which is here. And once we're in ventricle number 4, the choroid plexus, do you see this red over here? The choroid plexus of ventricle 4 makes and adds even more of this nutrient-rich CSF into this 4th ventricle that's over here, okay? So that's the steps of making the CSF and where it's going to travel to. It travels down, all the way down, okay? Then, step 6, CSF can take one of 2 routes, okay? Either from ventricle 4, the CSF can flow down this way through the central canal of the spinal cord, and half of it will, right? When you have 2 tubes, right, water will kind of split into 2 and go down both. So either some of it will go down this spinal cord here and feed the middle of the spinal cord, and some of it will go through a little hole this way, will flow through this aperture, this little hole here, to the 4th ventricle, that's found in the 4th ventricle, into this subarachnoid space. Remember, all of this is the subarachnoid space of the brain and the spinal cord, right? All this outer light blue. So some of it will go out over here to the subarachnoid space, just outside the 4th ventricle. And basically, in either case, right, the CSF will flow down over here, and then it will go outside. You don't see it, but once it gets to the bottom over here, right, it'll actually flow out into the sides over here onto each side. Here it will flow out and it'll go down this way. So in either case, right, CSF will then flow through the subarachnoid space and bathe the outer parts of the brain and spinal cord. Here, eventually at the bottom of the spinal cord, it'll turn out of the hole and it'll go into this subarachnoid space of the spinal cord. And here we know that we're already in the subarachnoid space of the brain, and we're going to be traveling all along here, right? So that's where it's going to go. And over here, we're going to go this way, all the way here. And eventually, everything kind of converges at the top over here of the brain. And by then, right, the CSF fed all these neurons over here, over here, over here, over here, right? All of these neurons, all of the spinal cord's neurons, right? All of the outer part of the brain's neurons, we fed them. But remember what this CSF is doing. It's giving oxygen, glucose, all these good things for the neurons, but it's also picking up their crap. It's picking up CO2, any waste that it has. So over here at the top, once you get over here, you fed all of these neurons and picked up all of their waste, right? So once you get to the top here, it's actually nutrient poor CSF, right? That is deprived of oxygen because all the neurons took them up. It's deprived of glucose, and it reaches these little, what we call arachnoid granulations. They're little granules, right, that are in the arachnoid matter. Remember, this pink is the arachnoid matter. The arachnoid matter does these weird little, they look like broccolis over here on the top, right? You see the little bumps. So once they get over here, the nutrient poor CSF reaches these arachnoid granulations and they drain into what we call this part over here, the dural sinus, because here in gray you have the dura mater, and this is a sinus. It's a space between the dura. So over here it'll go out into the dura, the dural sinus, and it will go down, 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 right? And it'll converge eventually and mix. Over here you don't see it, but this over here, this region, will actually then go down into your jugular vein and go back to the heart. Because again, this CSF, right, will then mix up. This bad CSF is deoxygenated. It doesn't have nutrients. It has waste in it. It'll mix with the blood. It's basically the same thing as blood, but without, it's picky blood. It's picky things that it picked out of blood without certain other elements, right, that it chose not to take in. And then it gives it to all the brain structures, and then it comes back nutrient poor, and it just drains back with the blood and goes over here, and then it goes down and returns down the jugular and will return back to the heart to get reoxygenated and get nutrients back into the blood. Okay, so all of this is the circulation of our CSF. So why does CSF need to drain, right? Why is it that over here you see your brain, you see our pia matter, you see the arachnoid matter with the little spider webs here, and here you see your little arachnoid granulation, right? And CSF, basically what it did was it was over here. It was flowing through these spider webs, right? And then it came here and it moved out into this dural sinus, right? So we had the dura mater here, the dura mater here, and you had a space here, the dural sinus, this big vessel here, right, where it will exit out. So why, before going through all that, why does CSF need to drain? Because neurons, right, during this whole process here, step 1, 2, 3, 4, 5, 6, and 7, right, during all those processes, as CSF is flowing here, it's feeding this tissue all around it, right? It's feeding the tissue that is all around it, and it's bathing the tissue, and it is giving neurons absorb the oxygen and the nutrients from the CSF, At the same time, these neurons, right, they do the opposite. All their junk that they have, they release it back into the CSF, all their CO2 and waste, they put it back into the CSF so that it could be brought and collected to bring back to the trash, back to the heart. So the used up nutrient poor CSF must now return to the bloodstream, right? So I kind of gave this away already, but where does the CSF exit? Nutrient poor CSF in the subarachnoid space. So once you get to the top here, in this subarachnoid space at the top, right, all this CSF will then go in the subarachnoid space and move into these arachnoid granulations, which are small granulation, sounds like a granule, a little grain, right? Grainy projections of the arachnoid matter. Here's a grainy projection out of the arachnoid matter. And the nutrient poor CSF can then cross, as I said here, you see that it's crossing the arachnoid matter to enter into the dural sinus, right? Over here it can't happen because the dura is here and it's very thick, but here in this granulation, it's an opportunity to exit. It's an exit ramp. So then it'll exit into the dural sinus and it'll mix with the venous blood. Okay, so it'll mix with all this venous blood that's coming back, and actually it will be directed to the back of the skull over here into this area. If I zoom into this area here, this is called the confluence of sinuses. I wouldn't worry too much about that. That's at the back over here. And so they all merge together, right? These veins come over here. This one comes this way, right? And all of that blood accumulates over here. And then, right, from here, all of it will exit out this way into the jugular, right? So this, from here, the CSF and the blood mixture flows into the jugular vein, which, as I said, it goes down the length over here in the neck, and it'll mix with more blood from other regions of the neck, of the, thoracic cavity, and then it is transported to the heart, to the lungs, to the kidneys. It's brought back to the lungs to be reoxygenated, and it's brought back to the kidneys to filter out all the wastes. And then actually it'll go to the GI system to get replenished with nutrients, and then it might end up going back to the choroid plexi and giving some nutrients to the neuron, to the CSF, to be distributed to the neurons. And so that's all we have for this nervous system lecture.